Hi, Florrie. Hi, Samuel. Hi, Mariam. Hi. Right, just while you're waiting for other people to kind of log on, the three that are here now, right, get a piece of paper and write down the method for quadrats. Okay, counting the number of daisies in a field. Okay, it's the one that's going to be on. Right, just while everybody's logging on, I've just kind of said it to the first couple of people. Just get a piece of paper, method for quadrats, right? Counting the number of daisies in a field. Okay, so counting the number of daisies in a field, right? Do yourself a method, right? And now think about it. Right from the very start, you've got an empty field. How are you going to get an estimate for all the daisies in that field? I'll run through it in a minute. Just repeat everybody. Just get a piece of paper, right? Write down the method for quadrats. Okay, for the number of daisies in a field. It's not going to be daisies, and they're probably not going to give you a field. I don't know what they're going to give you, right? But it is going to be a method, and there is going to be a calculation on it. Because there's very few required practicals in this that they can actually give you. Now think about it, right from the very start, right the way to the end, you've got to say how you're going to get exactly or an estimate of daisies. I'm going to give you another minute. Right, think about quadrats, daisies in a field. You've got a field, what are you going to do with it? Okay. You've got a quadrat, how big is it? Usually 50 by 50. How are you going to decide where you're going to put your quadrats? How are you going to decide? How many times are you going to put your quadrat down? When you've done it, however many times you're going to do it, what are you going to do with your result? Then you need to work out how you're actually going to estimate the number of days in that field. Another method. I'm going to kind of run through it, right? That one that you've written, that method, right? If there's anything extra stuff in it that you want to write, add, then do so. I'll come back to that in a second. Right. So Again, this PowerPoint is already up on Google Classroom. Okay, if you haven't got the Google Classroom code, right, then you need to email me straight at the end of this and I will give you the Google Classroom code. Okay, so there's your field, right? First thing you need to do is you need to create a grid, okay, and number it or letter it, okay? Because if you think about it, right, the standard way is throw the quadrat. Now, throw the quadrat is biased, okay? It is not accurate, it is not the way to do it. Randomize it. Okay, so what you're going to do is you're going to create a grid, right? And so you can have numbers at one side, you can have numbers along the other side, you can have numbers, letters, whichever you feel most comfortable with. Then what you do is you put the numbers in a random number generator. Okay, so a random number generator. So what it'll do, it'll spin and it'll choose two numbers. You then choose wherever you're going to go on that field based on that. And you place your quadrat at that point. Okay. Then you lay your quadrat on the ground. Okay, so there's no randomizing or well, there is random, it's biased, and you count the number of flowers if over 50% within that quadrat. And I'll show you a picture of that in a minute. Okay, then record it on a table and repeat steps 1 to 5 or 1 to 4 until you've recorded the number of chosen species in 10 quadrats. Okay, so again, be dead precise about how many times you're also going to do it. All right, so there's a kind of a big zoom in. All right, so on this right hand side here, this is then sort of clarifying a little bit what it is. Right, so if you look at that picture kind of on the right hand side, okay, then what it's got is it's got that quadrat there. You've got to tell the examiner how you're going to count the daisies in it. And the daisies in that is 2 because there's more than 50% of 2 daisies in there. If you look at this one on the top right hand side, okay, what there is potentially 4 daisies in there, but there's only that one and that one that are 50% in the actual quadrat itself. Okay, because that one and that one are not within that quadrat. Okay, so I'm going to go back to the method again. First thing you do, because again, this is going to be on your exam, right? First thing you do is create a grid. Okay, look at how big they've given it you, right? Use a random number generator to make the choice of each individual section random. Use the numbers and tape measure to locate the first position of your quadrat. Okay, so you've already set it out as a grid, then you need to find the right place. Lay the quadrat on the ground, so you're not throwing, you're not aiming for daisies, and count the flowers if over 50% within that quadrat. Record it on a table. Then, when you've chosen 10 quadrats, then what you'll then do is you will then calculate a mean. So it would be let's say there's there's 10 quadrats and your mean is 6.3. Then what you've got to do then is you've then got to work out an estimate of the number of daisies within that field. Okay, now what I've got on the screen there now is sort of a few examples of a potential kind of style of questions that they might ask you. Now, the most likely one is they're going to give you a field and they're going to give you some data and you've basically got to work out the number of daisies in a field, dandelions in a field, whatever it may well be. Okay, so this question here, I'm just going to go through it because I kind of know how important this one is. A group of students want to estimate the number of daisies in a rectangular field measuring 40. I'm just going to kind of draw it out on here. 40 meters by 25 meters. And you have a 0.5 by 0.5 square quadrat. So your quadrat is 0.5 by 0.5 meters. Right? Again, what I'll do in my head is I'll draw it out. Now, these are then some of the questions, so we'll just go through them, right? The student decide to throw the quadrat over their shoulder. Okay, just bear with me a sec. They decide to throw the quadrat over their shoulder. Okay, and immediately what you're then doing is you are introducing non-bias. Okay, hold on. Okay, so why throwing over the quadrat over your shoulder can lead to inaccurate estimate, right? It's all about bias, right? Because what you're going to do is you're going to have a bit of an idea in your head, aren't you, where you're going to kind of throw it. And you've decided also where you're going to stand in that field. But the whole point of doing it randomizing is to get rid of the bias. The second question on this then is describe how the students should determine where to place the quadrat to ensure their sampling is completely random and unbiased. That question is your method, right? So make sure you're starting right at the very start about gridding your field. Then what you need to do is you get your random name generator, number generator that I just mentioned. Lay the quadrat on the ground, count the flowers if over 50%, record, then you do a mean. Okay, right. Now, the question two, kind of the second part of it is the student placed 10 random quadrats. The total number of quadrats across all 10 quadrats was 64. Okay, so in 10 quadrats, there were 64 daisies. All right, then it says estimate a total number of daisies in the entire field based on the student sample. Okay, so if you've got 10 quadrats, 64 in total, then it is 64 divided by 10 equals 6.4 is the mean per quadrat. Okay, reasonably straightforward. Now, what I'm going to do now is I'm going to think about the size of my field. I've got 40 meters by 25 meters. So the area of that field is 1,000 meters squared. Okay, because literally all I've done is I've done 40 multiplied by 25 gives me 1,000 meters squared. Okay, now the bit that everybody kind of gets wrong is the fact that the quadrat is 0.5 by 0.5 meters. So what you got to do is you got to think that is 1 meter by 1 meter. Okay, how many of those little quadrats are going to fit in there? You will fit in 4, 1, 2, 3, 4 of those little quadrats into each meter. So therefore, if you've got 1,000 meters squared, that means there will be 4,000 quadrats that can fit into that field. If you've got 4,000 quadrats in that field, to work out your estimate, okay, because remember it's an estimate, it's 4,000 multiplied by 6.4 gives you your answer. Okay, now again, when you're actually doing this in your actual exam, go through each of individual steps, right? But the biggest thing that everybody always gets caught out on is the size of the quadrat. Okay, other permutation of it could be they could give you a 0.25 by 0.25, which would be like 25 centimeters by 25 centimeters quadrat. Think about how many of them would go into a meter square. So if I've got a meter square, you would then be able to get 4 across the bottom and you get 4 up the side. Therefore, the total in that one will be 16, right? So watch out for the size of the quadrat. So half a meter by half a meter, you'll get 4 in a meter. 0.25 by 0.25, you'll get 16 in a meter. Right? Now, question number 3. The teacher informs the students that the true population of daisies in the field is significantly higher than their sample estimate. Okay. Now, it could be significantly higher or it could be significantly lower. Now, it says there then suggests 2 reasons why their sampled estimate might have been unrepresentative low. Okay, so what you need to do for that one is you need to think that the sample size is small. Okay, because if you think on that field example I'm giving you, there's 4,000 quadrat possible placements and you've done 10. So therefore, the sample is very small. So what you could do is do 100 samples. What you're going to do then is you're going to get it a lot more accurate. Another way in which it could be wrong could be miscalculate or human error. Okay, because remember when I was talking about it before, you've got to make sure that 50% are within that square. You might not count like that. You might be counting if they're all in there. Okay, and also the random generator might not be random enough. Okay, so for example, in my classes, what I do is I use a random name generator to choose people in class. And if I've got 36 kids in my class, right, sometimes I can get the same person called 3 times on the trot, right? So if for reasons why your answer could be wrong, the main ones are the sample size is too small, you might miscalculate, and also the random generator may well not give you random enough answers, right? So, and the final one then is state one way the student could modify the results to make their final estimate more repeatable and stable is increase the number of samples. Right? So, quadrat one, right? Highly likely. Right? Now, whether I get round to it or not, at the end of this PowerPoint, there is 2 other quadrat GCSE style questions. And what I've done is I've made them a little bit different, where one of them is a field that is circular and another one is a field that is L-shaped, right? So you never know what they're going to do. So at the end of this, if I haven't got to it, then just have a look at those 2 questions. The answers are there, right? And have a go at doing them. Okay. Right. Now, the other part of the quadrat one, so most likely it's going to be that one, but they could also do the transect. Okay. Now, the difference between the kind of counting the daisies is where you're actually getting a number. The transect is you're more looking for a pattern. Okay. So what I've got on here is I've got my tree. Okay, so I've got my tree. All right, and then what you do is you then lay a line. Sorry, you then lay a line. Pen back on. Okay, you lay a line using a tape measure, let's say 30 meters. Then what you do is you put a quadrat at the start of your tape measure, leave a gap, and then put another one. Right, so then what you've got, you kind of got a spacing of quadrats. If you're asked for daisies again, what you've then got to do is you've got to count the number and you've got to mention the fact that it's only going to be 50% within that quadrat. And then you kind of go through exactly the same method. But what they're normally going to do is they're going to normally ask about light intensity. Okay, now often again, they'll tell you the light intensity equation, but then I've written it here. Light intensity is 1 over the distance squared. Now, we talked about this in biology paper 1. Okay, so light intensity is 1 over distance squared. And again, remember that if you double the distance, you quarter the light intensity. Okay, that is a very, very, very important fact if they start talking about light intensity. So to say it again, if you double the distance, so double the distance, the light intensity quarters. All right. So what you again do is you lay the quadrat down every kind of 50 centimeters, then leave a 50 centimeter gap. Okay. And you count the number of plants in size. And at each point, you also measure the light intensity. Record your results on a table and continue to place the quadrat until you get to the end of however long it is. And it could be 50 meters or 5 meters, 30 meters rather. Right? Your table could look a little bit like that, where you start and then 5 meters along, 10 meters along. Again, they're going to tell you the distance between each individual quadrat. You put the number of plants in a quadrat and then you do your light intensity. And that is the inverse square rule. All right. And what you're going to do on this one is you're going to be looking for a pattern. So what they could do is they could give you a graph like that, right? And that could be numbers, right? And then all of a sudden what happens is it dips for some reason. And you've then got to maybe, based on a picture that you got, why does it dip? So you got 100 flowers, 100 flowers, 100 flowers, and all of a sudden it goes down to 20, and then it goes back up to 100 again. You've got to work out what that could be. It could be a path. Okay. Or it could start very low and go high, right? And what it could be under a tree. So then you've got to think, what's that tree taking out of the ground? It's taking the light away, right? It's taking the minerals away. It's taking the water away, right? So just study the picture and study the diagram that they give you, and then you'll be able to work out all the information. Right, the second required practical is your reaction time. Okay, I'll go to the sort of the written one like this. Now, again, you've got to be precise. All right, you've got to be dead precise and you've got to chuck numbers in. So, person number one, think about you're working with somebody else. They put the forearm of their dominant arm across the table. Okay, so forearm of your dominant arm across the table with your hand then sticking over the edge. Person two holds a ruler between person one's thumb and forefinger. All right, a variable is the fact that your thumb and forefinger are 2 centimeters apart. Okay, because what you got to do is you've got to keep everything controlled. When you do it, person two lets go of the ruler and you record the number onto a table. So it might do 27 centimeters. Then, weirdly, because it's such a tiring experiment, you then have to have a minute's rest. Repeat it 5 times, 10 times. Then, to actually convert your 27, or whatever your mean is, you need a conversion table. And a conversion table looks a bit like this. Now, you might get a conversion table, you might get a graph. Okay, so if you then get a graph, then what you've got to do is you've then got to look at where your mean is. So if it's 27 centimeters, you go across to 27 and up to your reaction time. Okay, but often they look like this. So if I think 27 centimeters, that's my mean there. So my reaction time is 0.23 seconds. Okay, right. Now, what they're also going to do, they're also going to chuck in a comparison. Right? They're going to say, you've done it with your normal right hand. Compare the results for your left hand. You might have a female partner. Compare the right hand of a male and the right hand of a female. You might have, you've got your right hand and then you have a drink of coffee for caffeine, right? And again, look out for what they're kind of testing, because if it is something like caffeine, you've got to give 30 minutes for the coffee to be absorbed, right? So again, it's very difficult to predict exactly what it is, right? But if you go through the method I've just told you, right, and then look out for what they are doing the comparison. All right, so variables, don't mess in class. So don't mess. What are we measuring? We are measuring our reaction time. We're not measuring the distance on the ruler. We're measuring the time for your reaction. What are we going to change? It could be the person. It could be the hand. It could be caffeine. Okay? It could be one eye. Okay? Control variables, the ruler must stay the same. The person dropping the ruler must stay the same. The distance between your fingers must stay the same. And another thing that I didn't mention before is you should always be looking away from the ruler, right? So when you're actually doing this, okay, if I was doing this on my desk here at the moment, I wouldn't be looking at the ruler, I'd be looking at a point on the wall, right? Because the idea of your reaction time is your peripheral vision, right? Now, hopefully at the end, I'm going to go through these again. Okay, right, now, biology is more wordy. Okay, so what I'm going to do now is I'm just going to remind you of our words. All right, so first thing is read the full question. Okay, now hopefully after 3 exams, what you're going to be able to do now is you're going to be able to understand that every single little clue, right, or every single word in that question means something. Define is your definition, right? Main one that you're going to get in this is probably homeostasis, and we'll go through that in a minute. Describe, facts, calculate, do a sum. If it says use the graph, you're going to use the graph. If it says use figure 4, you use figure 4. If it says use figure 4 and figure 5, you are going to be doing something with both of them. Explain so, suggest, give an idea, compare is however, and evaluate but, and also because. Right, so again, look for that keyword. Right, now what I'm going to do now is I'm going to start going through our theory, okay? Because obviously our theory, right, is what you then kind of need to know. Right, now, comparison, endocrine and the nervous system, right? So what I'm going to do is I'm going to go through kind of the main comparisons. Right, I'll put my camera down a bit. The main comparisons. So endocrine system is your hormones. Nervous system is obviously your nerves. So each system is made up of different things. The endocrine system is made up of your glands, right? And in a minute, I'm going to go through a load of different glands and a load of different hormones. The nervous system is the nerves, the brain, and the spinal cord. And it could be the CNS and the PNS. Central nervous system, your brain and your spinal cord. The PNS, your peripheral nervous system, right, is kind of all the stuff that's to the sides, right? So when I'm sitting here now looking at my laptop, I can see out the sides of my eyes, that is the peripheral. The message, okay? So the type of message, electrical impulse, chemical hormone. Speed of transmission, very fast. You're talking about fractions of a second. Slower. Length of the effect, short, because literally you send a nervous impulse, and then what you do then is it then happens. Length of time, long. Okay? Because again, if you release a hormone, it takes a minute and a half, 2 minutes to go around your body. Okay. Now, another reminder or exam technique. If it says compare, right, make sure in your answer you're also using your compare words. So where I've got length of effect and I've written short, that's rubbish, right? You're going to write shorter. Okay, again, don't forget that keywords. And you're also going to write longer, right? So when you say comparisons, remember your ers on the end as well. Okay, so that's the endocrine and the nervous system. Right, we'll start off with a little bit to do with the nervous system. Okay, there is your reflex arc. Okay, so reflex, I could throw something at anybody that's kind of here tonight, right? Then you'd move your head out of the way, right? That is a reflex. So it starts off with a stimulus, right? Often a bee. Receptor, if we're talking about a bee, is your hand. It then goes down the sensory neuron to the relay neuron, and the relay neuron is in your spinal cord, to a motor neuron, to an effector, and then a response. Okay. Now, additional things that could be kind of included in that is, and I'll run through these in a minute, in between the sensory neuron, the relay neuron, is your synapse. Okay, and again, I'm going to talk about synapses in a minute. An effector can be a muscle, or, can't spell muscle, or a gland. Okay, so they are then, okay, they are your effectors, is a muscle or a gland. Okay, and then I've got another diagram here where we've got the receptor cell, impulse goes up from a pin, and it goes into up the sensory neuron, to the relay neuron, to the motor neuron, to the effector. Right? Now, don't forget as well that in a reflex, the brain isn't included at all. Right? Hence why it's a lot quicker. When it gets to the relay neuron sends a message to the brain just to say what's happened. Okay, so if I threw a pen at any one of you, you'd move your head out of the way of your reflex, but then because that's happened, you would then send the message to the brain, and then your brain would know that Mr. Leek's thrown a pen. Okay, but it's not involved. So that is a reflex arc, reflex pathway. Okay, right. Now, we've also got a comparison with the nervous system of voluntary and involuntary actions. Okay, so you've got to think voluntary actions are things that you are deciding what you do. Involuntary actions are actions that you have basically no control over, right? So a voluntary, there's no awareness. Cerebrum, now I'm going to put cerebrum, right? And that is more kind of for your separates, right? But anybody not doing separates, then you need to think that basically voluntary actions are controlled by the brain. They are slower because you're then using your brain to actually make something happen. And examples of voluntary actions are speaking, hearing, writing, walking. Okay? Anything that you are kind of doing, you're consciously doing, that is a voluntary action controlled by the brain, and you have awareness, but it's slow. An involuntary action, right? So medulla oblongata, that's the separate science bit. For everybody else, basically, it is your spinal cord. Okay. Involuntary actions are very fast. Okay. And again, it's things that you can't control, like your heartbeat. Peristalsis is the movement of food down your esophagus. Respiration, okay, and anything else like your breathing and things like that that you basically cannot control. Right, synapse. Okay, now down the right hand side of that screen, right, if you want to kind of do it in huge amounts of detail, right, that sort of goes through it. But what I'm going to do now is I'm just going to kind of show you what happens with the synapse, right? So what we've got here, let's call it the sensory neuron. So therefore, we'll call this the relay neuron. All right. And what happens is a nerve impulse comes down the sensory neuron. Change the color. Okay. A nerve impulse comes down the sensory neuron. And what it does, it stimulates the release of a neurotransmitter. So a neurotransmitter, right, which is written there, from the presynaptic membrane. Right. So an impulse comes down the sensory neuron. It stimulates the release of a neurotransmitter from the presynaptic membrane. Okay, presynaptic meaning before synapse. The neurotransmitter diffuses across the synaptic cleft. Okay, so synaptic cleft is the gap in the middle. And then there are receptors here. These kind of teeth looking things are then receptors. If it is detected, the impulse then carries on down the relay neuron. Okay, so that's quite a complicated one. And what I'm going to do is I'm going to take it off. Okay, all my ink stuff. All right. And now, just going to run through it again. Impulse comes down the neuron. Stimulates release of neurotransmitters from these kind of little bag things into the synaptic cleft. These are then detected by receptors on the relay neuron. If there is enough detected, the impulse carries on. If there isn't enough detected, the nerve impulse stops here. Okay. Now, if you want to go through that in a bit more detail, the details of it will tend to come up more up on the higher paper. Okay. But go back on this PowerPoint afterwards, and down the right hand side, it kind of goes through it again, right? With all the kind of the pre and the post. Right. Homeostasis. This is now your definition. Okay. So if they're going to ask a definition, pretty damn good chance it's going to be this one. Okay. So homeostasis maintains the optimal conditions for enzyme action and all cell functions. All right. So your blood glucose, your body temperature, your water levels, okay, all the things that must stay the same in your body, that is then called homeostasis, and it is to maintain the optimal conditions for enzyme action, right? So it's all about the enzymes, right? So think about in previous ones where we've talked about enzymes and pH, right? If you get too hot, they denature. If you get too cold, they slow down. If your pH changes, they denature. If your pH goes down, they denature. So that definition needs to be there. Homeo pretty much means humans. Stasis means stays the same, stationary. So homeostasis is human stationary, and it is to maintain the optical conditions for enzyme action. Okay. Now, the three that I'm going to kind of talk about a little bit, now the last two we don't really need to do very much, is blood glucose concentration. Again, you don't talk about glucose, you talk about blood glucose concentration, body temperature, and water levels. Now, the only one that combined that you need to know any detail about is the blood glucose concentration. So we'll do that one in a second. Okay. Right. Now, before we start talking about the blood glucose, we're just going to go through some of the hormones or some of the glands that you could get asked about. Right. So endocrine. Now, if I'm just going to sort of name and sort of talk about where they are first, and then we'll go through a little bit more detail. So the pituitary gland, right, is the master gland, and it's basically in kind of the base of the brain. Best way to find it, if you kind of go straight in between the eyes, right, or slightly above the eyes, right, and you go back in about sort of that far, right, maybe about 7 or 8 centimeters, it's about the size of a pea. That is your pituitary gland. Your thyroid gland is in your neck, right? It tends to look a little bit like a butterfly, right? So when you're ill, what the doctor does, the doctor squashes around your neck because they're trying to see if the thyroid gland is swollen. The pancreas, we'll talk about that in a minute, which is insulin. The adrenal glands that are on top of your kidneys. So if you think, right, underneath your ribs at your back, right, just underneath the ribs at the back, that's where your kidneys are. And your adrenal glands are smack bang in the middle of your body. Then you've got testes, testosterone, and then you've got the ovaries for the female sex hormones. Okay, now I'm just going to kind of go through a number of different ones. Okay, firstly, the pituitary gland. Okay, so the pituitary gland has a number of different hormones that are secreted. Now, these are them. Okay, so what I'm going to do is I'm just going to kind of talk a little bit about them, about what they do. All right, so the growth hormone. Right, so the growth hormone, what it does is it regulates growth and development. Right, and what it does, it basically tells you how big you're going to be. So your arms, at each end of the arm, right, or the bones in the arm, that is then when growth happens. Right, so my arms have obviously stopped growing, right, but if I was still growing, kind of at my elbow, that is then when growth hormone stimulates the length of my arms growing. There's a medical condition called acromegaly, right, and that is then when certain people have cancers or tumors on the pituitary gland. And they are those people then are like 8'9" tall. FSH and LH are female ones. Okay, so what you've got is, if I just draw an ovary, okay, so what you've got, all the ladies, right, you've got literally tens of thousands of follicles in your ovaries, right, and you're born with 100,000 follicles, all right. What FSH does is produced by the pituitary gland, goes through the blood to the ovary, and it makes one of those follicles stimulate it to mature to become your egg. Okay, and as the FSH stimulates the follicle to become an egg, then LH, which is luteinizing hormone, gets produced, and luteinizing hormone causes ovulation. Okay, so those two produced by the pituitary gland, and literally what they're doing is they are then maturing an egg and then ovulation. Right? Now, the other two, thyroid stimulating hormone, TSH. Okay, thyroid stimulating hormone is the hormone that stimulates the thyroid gland. So the pituitary gland produces a hormone to stimulate the thyroid gland, and we'll talk about the thyroid gland in a minute. ADH, right? Combined, you only need to be aware of what it does. Okay, so ADH controls the amount of urine that you produce. Okay, and it's called anti-diuretic. Diuretic is something that causes you to wee. Okay, so for example, if you have a big cup of coffee, right, what that cup of coffee is a diuretic, and it makes you wee. Right, now, glucose. Right, again, we're going to talk a little bit about some of those others, okay, because there's lots and lots that we need to do. Now, remember your definition of homeostasis. Okay, maintaining the optimal conditions for enzymes to work. Right, so the way that it does it. Straight after this, you go and get a cake. Your blood glucose levels rise. Your pancreas detects the fact that the blood glucose has risen. Your pancreas produces insulin. Your insulin then takes up the excess glucose and stores it as glycogen. All right, so what you've got to do now is you've got to think now there's a few stupid words that are included in this. Glucose, oops, and glycogen. All right, so I'm just going to say that one again. You eat a cake, your pancreas detects glucose levels go up. You then produce insulin. The liver takes the glucose, because it's gone up, stores it in the liver as glycogen. So glycogen is a very, very, very high energy compound in the liver. Now, if you are asked about this, make sure you then say, you then return to homeostasis. Okay, so your 3 marker then will be insulin to glycogen back to homeostasis. The second possible route is you run up the stairs, you do a load of exercise. The pancreas detects that the glucose levels have gone down. They then produce a hormone called glucagon. So I'm just going to write down glucagon over here. Glucagon. Okay, so glucagon is a hormone that does the opposite to insulin. That makes the liver break down the glycogen and release glucose. So therefore, if glucose is then increased, your blood goes back to homeostasis. Right? Now, the reason why I've written those 3 G words down the side is you can't get them confused, right? Because basically it says in the exam marking scheme that if you use one in the wrong place, you don't get marks for it, which is kind of obvious, I guess. So glycogen is the store of energy in the liver. Glucagon is the hormone that does the opposite to insulin. Okay. Right, diabetes. Right, this is a great comparison, or it could be just an explain. Right, so type 1, you are born with. Okay, so basically anybody that's got type 1 diabetes, you have it from as soon as you pop out your mum. Right, often detected within kind of about the first sort of 2 to 6 months. The only treatment for it really is regular insulin injections. Okay, so there's a lad in one of my year 11 classes, he's got diabetes, and he's got a little detector attached to his arm, and if his blood glucose goes wrong, then a alarm goes off, and he then has to either go and eat something or get some insulin. Okay, so you're born with it. Type 2 is all about life choices. Okay, so think about the life choices that you make, right? You guys are all young, fit, and healthy. Okay, I'm not, right? So if I've made then life choices in my time, like eating too much, not exercising, smoking, drinking alcohol, right? They are potential life choices that could bring on type 2 diabetes. The main one is obesity. Now, luckily, I'm not obese. Okay, so obesity is one of the main kind of risk factors. Treatment is you change your diet and you increase your exercise. Okay, so type 2 diabetes is usually brought on by obesity, but it's also to do with smoking, alcohol, lack of exercise. Okay, treatment for it is eat less, stop smoking, drink less, exercise. Okay, so type 2 is all about choices that you make in your life. Right, now I've just chucked in accuracy here again, right, because accuracy is just a word that comes up all the time. Okay, so accuracy, repeat, right? Often I'd say 10 times. Then calculate a mean. Second way of increasing accuracy, increase the resolution, right? And I'll often say, use something digital, okay? And if you increase the resolution is to increase, or resolution means being able to distinguish between 2 points. So accuracy, use something digital thermometer, right, to increase the resolution. Right, adrenaline. Okay, now I like the adrenaline one. Okay, it's kind of quite interesting. Okay, now I have tried to predict adrenaline being on somebody's exam paper for years and it's not been on. Okay, so on there I've got 8 effects of adrenaline. Now, what I suggest you do is maybe even on a little piece of paper, right? You then think of maybe 3, maybe 4 effects of adrenaline that kind of make the most sense to you. Okay, but I will say you need to include breathing rate. You need to include heartbeats faster as 2 definites. So this could be a good explain, right? What I'll do is I'll just kind of talk through them, and then you then think about maybe the 3, maybe the 4 that you want to kind of note. Right, a tiger walks in the room. Okay, so the effects that you do is your pupils widen. So you can get more light onto your retina. Your heart beats faster and deeper, so you can get more oxygen and glucose to your muscles for respiration. C, glycogen, right? We've just talked about that. Glycogen is then in your liver. So glycogen's hydrolyzed, so you then have more glucose in your blood. D, more blood delivered to the brain, so you become more aware, right? If you think about it, when you get scared, right, what happens is you do become more alert. E, more blood delivered to the skin, so you can lose heat. F, less blood delivered to the gut, right? A tiger's about to eat you. Do you really need your intestine doing its job? G, breathing rate increases, so you can get more oxygen into your blood to go to the muscles for respiration. H, blood flow to the muscles is increased, so there is more oxygen and glucose in the muscles for respiration. Okay, right, so adrenaline, gut feeling, but it's been a gut feeling for years, right? So think about 3 of them, but then also think about why. Right. That's then just kind of another kind of adrenaline picture that I kind of found, right? You got to remember that adrenaline is a bit like fight or flight, right? Tiger walks in the room, you either fight it or leg it. Okay, I'd advise leg it if a tiger did walk in, right? And just think about the whole of your body, right? About what could potentially happen. Right, now here's another question that they could talk about. They could talk about negative feedback. Okay, I always find this one a little bit tricky to kind of explain, right? So if I just kind of read it out, right, and then what I'll do is kind of a little talk about it. So negative feedback is a regulatory mechanism, right, in the endocrine system where an increase in one hormone, so if you increase one hormone, what it does is it triggers a response that reduces more of that hormone to be made. Okay, so negative feedback is where you have a rush of a certain hormone, but that hormone then makes a mechanism where it tells the gland to produce less of it. Okay, so it's a bit like our insulin. So You eat a cake, your body produces more insulin. The negative feedback is then where your blood glucose goes back to normal. So that is the negative feedback to stop you producing any more. Okay. Homeostasis again, maintaining the optimal conditions for enzymes. Right. Now, that's again another example of negative feedback. I'm not quite sure how they're going to ask about it. Right. So what happens is conditions in the body change from a set point, right? You eat glucose. Your change is detected in the pancreas. Your pancreas produces insulin. Then what happens is your glucose decreases. Therefore, you don't need to produce any insulin anymore. The negative feedback is where your hormone resolves a problem and it prevents anything else then being made. Okay, right, menstrual cycle. Okay, so what I'm going to do is then that's a good diagram, right? We've already talked about two of the hormones, but we're going to talk about two of the others. Right. So if we talk firstly about FSH, right? I drew a picture of the ovary before with 100,000 follicles in it. All right. So FSH is produced to mature a follicle into an egg. Okay, and you can see this here. Right now, just for the lads, right? Because obviously they're going to be very unaware of this entire cycle. Okay. 0 or day 0 is when the bleeding stops. Okay, so when the bleeding stops, then basically the menstrual cycle then starts. Ladies will then be producing FSH to get a follicle to be matured. When the follicle is matured, you can see that yellow line then decreases because then you don't need it anymore. Once that follicle is then matured, the next main hormone for us to talk about is then the LH. LH is then this one. So you've now got a mature follicle. All of a sudden, there's a peak in LH, literally over 4 days, ovulation. That is then when the egg pops out of the ovary. As soon as it pops out, LH decreases and you don't need it anymore. So you're literally producing LH for 4 days in a menstrual cycle. Now, the other two hormones that are in there, okay, are estrogen and progesterone. All right. So estrogen is to kind of build up. Estrogen is to build up the uterus. Okay. So what it does, it builds up the uterus and kind of gets it ready for any implantation. Progesterone maintains the uterus. Okay. So the uterus gets kind of built up by estrogen. Once it's built up, progesterone maintains it, so it keeps it optimal for when there is any egg implantation. And as soon as the body realizes at kind of around 22 to 24 days, there is going to be no egg, progesterone decreases, and that is then when period starts. Okay. Now, also remember, menstrual cycles vary. All right. Some women have menstrual cycles every 6 months. Some have them every 28 days. Okay. Some have them every 14 days. Okay. It's all kind of to do with regulation, right? But generally, it's 28 days. Okay. Right. Hormones again, right? We obviously just talked about estrogen, right? But estrogen is the main one or main job of estrogen we just talked about is sort of building up the uterine lining, okay? And it is then involved in kind of ovulation. But from a kind of early point of view, estrogen is the hormone that brings on secondary sexual characteristics, okay? Like breast, like hips widening, like ovaries becoming more mature, like the starting of the menstrual cycle. Testosterone in lads, made in the testes, And what testosterone will do, that will bring on male secondary sexual characteristics, which is more facial hair, deeper voice, widening of shoulders, and production of sperm. Right, so you asked about testosterone, deepening of the voice, widening of the shoulders, production of sperm. Okay, they are then the male secondary sexual characteristics. Right, another thing to do with fertilization. Okay, in vitro fertilization. Okay, right. So again, it's all to do with kind of the female hormones. Right. What I'm going to do is I'm just going to mention the thing in a second that everybody always forgets about. So in vitro means in glass. Okay, so vitro is just another word basically for glass. Okay, so in a very kind of sort of random kind of way, right? What happens is mother produces eggs. Okay, can be 10, can be 4, can be 12, right? When the eggs are produced, what they are then is they are fertilized by dad. Okay, so dad will then give some sperms, and literally what happens is they'll have a petri dish, which is like a little glass bowl. The eggs are in it. They'll put the sperms in. The sperms will swim around within all those eggs, and then what they'll do is they'll fertilize the eggs. Once those eggs are fertilized, they will then develop into embryos. Okay, so an embryo is sort of the first kind of 10, 15 days of the life of a baby. It's kind of an embryo. Okay, then the doctors will put 2 embryos back into the mother. Right? You'd imagine they'd put 1, but they don't because they put 2 in to double the chance of implantation. Okay. Now, again, with IVF is you've got more chance of having twins. Okay, because obviously if you've got 2 in and both of them kind of implant, then what can happen is you can get twins. Okay, now the bit that everybody forgets, and I've kind of put in this now as orange, is before any IVF has to happen, mum is given strong doses of FSH literally over about 2 months. Okay, because it doesn't just go in and get the eggs taken out. What happens is there's a kind of a good 2 months of kind of doctor input, right, to get those eggs produced. So doctors give FSH. And then what the woman will do then in both ovaries, she might produce 5 eggs in each ovary. Just as they're about to then sort of take those eggs out of the ovaries, they will then, or the doctors will give LH. Okay, so remember the 2 hormones. FSH is follicle stimulating hormone. So then you've got your 10 mature eggs. Then they're given LH, which then prepares them or gets them ready for ovulation. And as they're getting ready for ovulation, they put a tube in, right, a big needle in, okay, to where the ovary is, and they literally suck the eggs out. They then, and I go back to my original, they then get 10 eggs, 5 eggs, 12 eggs, don't know how many. They fertilize them using sperms in glass. You then get an embryo, and then they reimplant 2. Okay. Now, the other 8 embryos are usually frozen. Okay, so what they are is they're put in liquid nitrogen and they're frozen just in case the woman is unsuccessful. And then what they've got is they've got some ready-made embryos that then can be reinserted. Okay, it's like my sister. My sister's what, 57, something like that. She had IVF when she was about 30. 20 years later, she had a phone call saying, right, we've got 10 eggs, right, 10 embryos. Do you want them? Right, and they were literally asking at the age of 50, do you want to have another baby? So she said, yes. What an idiot, right? But luckily, none of them implanted. Okay, so then they are frozen. Right, now, big problems with IVF. Okay, so this is, again, it's kind of something you have to think about. Emotional and physical distress. Okay, so if you think if a woman and a man, right, what they're doing is they've probably been trying for a long period of time to have a baby, right, and all of a sudden they've then got to go down the IVF route, that means they're pretty desperate. So emotional, physical stress of actually going through the IVF process. Success rates are not high. Hospitals will often say 50% success rate. Okay. People move house to get as near to a hospital with high success rates. Okay. I don't live far from a hospital here, right? And if this had a good success rate in IVF, right, people would actually move to be near this hospital to get those success rates. Some hospitals will give one chance, some will give two chances. Okay, and it's not like about £8,000, £10,000 to have a go yourself. And there is multiple births and obviously risk to mother and baby, right? Because mother is going through a number of kind of different sort of scenarios. Okay, right, now on to a bit of genetics. Okay, so what I'm going to do is I'm going to go through some of these genetics things. Okay, and we'll start off gamete. Right, gamete is a sex cell produced in meiosis. Okay, so your gamete is a sperm and an egg, right? And they are produced in meiosis. Chromosome, long chain of DNA found in the nucleus. So what you've got is you've basically got 46 of them. Okay, so they're 46 chromosomes. Women have got 46 X's. Men have got 45 X's and one Y. The gene, if I was then going to zoom in on a chromosome, a gene would be a little tiny section, right, of the genetic information that then gives you a characteristic. So it could be size of your nose, shape of your ears, okay, color of your eyes, color of your hair. That's a gene. And an allele is a different form of the same gene. So that there, that I'm then going to highlight in red, that there is a gene. Okay, and every single one of you, near enough, has then got different alleles. Okay, which means your ears are going to be slightly different, your nose is going to be slightly different. So an allele is an alternative form of the same gene, a bit like an isotope in chemistry. Right. Keywords again that they're probably going to mention. Right. So what we've got is we've got dominant. So dominant is the stronger gene or stronger allele. Usually it is the capital letter. Recessive is the weaker of the alleles. So it's usually, well, it is a small lowercase letter. Homozygous. Homo means two of the same. So if I've got big R and big R, that is homozygous dominant. Heterozygous is where you've got two different ones, R and little r. Okay, going back to homozygous, if I've got r, and little r, that is homozygous recessive. Okay, again, remember kind of your wording that kind of goes with this. Last two at the bottom there, I'll just clear all the other rubbish that I've then just written on it. Okay, so last two at the bottom there, genotype. Genotype is the alleles that you've got. Right, so what I could do is I could then take a blood sample from one of you, right, and I could get a big full long list of every single gene inside you that says exactly what you're like. That is your genotype. Your phenotype is what you physically look like. Okay, so the color of your hair, your eyes, the shape of your ears, whether you can roll your tongue or not, right? Because rolling tongue's quite a good one because it's one of those ones you can either do or you can't do. Right? And however much you practice, if you can't roll your tongue, you're never ever going to be able to do it. Right? Ears as well. Ears is another kind of standard one, right? Because people either have ear lobes or they don't have ear lobes, right? There's kind of no in between. So you either have a little dangly bit, right? Or it goes straight into the side of your head. Right. Now, the Punnett square. Okay. Just to again talk about percentages a little bit. Okay. So what we've got is we have a father and mother both carry a gene for cystic fibrosis. Now we'll talk about cystic fibrosis again in a little bit, okay? Because it's one of those genes, one of those comes up. Right, this is then the Punnett square for it. So heterozygous. Oh, why not? Okay, heterozygous dad, heterozygous mum. Okay, then Punnett square, you do big C, little c, big C, little c. Right, you all then know how to do Punnett square. So then what you're going to get, you're going to get big C, etc., etc. The last percentages, right? And you're just going to look out what they are actually asking for. So a carrier means they haven't got it, but they have got the allele. So carrier, carrier, because it's 2 out of 4, 50%. Cystic fibrosis. Cystic fibrosis is recessive, and it needs to be recessive homozygous. That's that box there, 25%. No disease. Homozygous is then 2 of the same. Heterozygous is 2 different. Think about your percentages. Right. Now, there's 3 main Punnett squares that they're going to talk about. The first one is sex-linked, right? And it's explaining why there is 50% males and 50% females in the world. Okay. So women, what they can do is they can produce only female eggs. So they can only produce gametes that are XX. Men can produce either female sperms or male sperms. So at the end of the day, the men actually choose what is the gender of the baby. You do your Punnett square, 50% male, 50% female. Okay, so if they ask anything about why is 50% women, 50% men, okay, then this is then kind of your evidence, which is then your Punnett square. Now, the 2 diseases that are actually written on these syllabus. Okay, so first one, polydactyly, well, cool. Okay, poly means lots of, dactyly means fingers or toes. Okay, so dactyle, right, are your kind of fingers. Now, the key to this is not the fact that it looks quite funny, right, is the fact that it is a dominant allele. Right, there's very, very, very few kind of inherited disorders that are dominant, because that usually means you're going to get it. So looking over on the right hand side here, I've got 2 Punnett squares, right? 2 heterozygous parents. So because dad has got the big P and mum has also got the big P, that means they've both got 6 fingers. When they reproduce, 75% are going to have 6 fingers. The little p, right, will be the weird one in the family because they will have 5 fingers on each hand. And they'll probably get kicked out and sort of ostracized and adopted because they've only got 5 fingers on each hand. Okay. If you've got 1 heterozygous and 1 homozygous, so dad doesn't have 6 fingers, but mum does, it's then 50% chance that they're going to have 6 fingers. Okay, but I'll just repeat it again. The key to polydactyly is the fact that it is dominant. Okay, because it's unusual. Right, cystic fibrosis. Cystic fibrosis is recessive. Okay, so therefore that means that both parents must carry it for a child to have cystic fibrosis. Okay, because it is the little c, not the big C. All right, symptoms of it. What you have is you have thick mucus. All right, so if you think about it now, you know when you get a really bad cough and you kind of got that stuff that's stuck in the back of your throat? Cystic fibrosis is like that all the time. Okay, so what it's like is it's like thick mucus all the way down the trachea into the lungs. They also have thick mucus in the intestines. Okay, so they're going to frequently get coughs, colds, sort of infections quite regularly. Treatments, they have a lot of antibiotics. Okay, and what they'll also do, they'll also have certain massages. Where what they might have to do, they might have to lie on the front, right, and they kind of get battered on the back to release the mucus to then get the mucus out. They're also supposed to eat very, very high fat food. Okay, so it'd be good for them to go and have a KFC every night. Okay, so because the intestine is then kind of full of mucus, right? If you eat a high fat food or high fat diet, it slows down your digestive system. So the idea of slowing down the digestive system of somebody with cystic fibrosis is to give the body more time to absorb all the goodness. Okay, so again, cystic fibrosis is recessive. Okay, right, now sexual and asexual reproduction. Okay, could be a compare question. So there's kind of two questions that kind of go through my head that there could be to do with sexual and asexual reproduction. One could be a comparison. The other one could be advantages and disadvantages of either one. All right. So what we've got to do now is just got to think about kind of sensible answers that we could put. Okay. Right. So at the top there, I've got asexual and sexual. Asexual is where there is one parent. Okay. So think about your bacteria. So your bacteria, right? There's obviously not a mummy and daddy bacteria. There's one bacteria, and then it reproduces by mitosis in about 30 minutes. One parent. In sexual reproduction, there's obviously two. Cell division, mitosis, meiosis. Okay, now I know combined you don't need to do very much with meiosis. Okay, where separate you do need to do more with meiosis. Okay, so mitosis is where it just splits. Meiosis is where you're combining a sperm and an egg together. Genetically identical. Mitosis, variation. Okay, so think about your comparisons here. Right? Asexual reproduction has one parent. However, sexual reproduction has two parents. Asexual reproduction uses mitosis. However, asexual or sexual reproduction uses meiosis. Asexual reproduction produces identical offspring. Sexual reproduction produces variation. Okay. I'll just skip down to the fourth one down. Number of offspring, large numbers. Okay, because in mitosis, what they're doing is they're splitting all the time. In sexual reproduction, very small numbers. Asexual reproduction, fast. Okay, right? Imagine if you could have a baby in 30 minutes. Okay, that looked exactly like you. Sexual reproduction, slow. Okay, so think about your howevers if you're going to get a comparison question on that. Right. Okay, moving away from the all that stuff, right? We'll talk about eutrophication. Okay, so eutrophication, right? What it'll be foundation question. It'll be an idea about what it is. Higher question, right, is then where it's going to need to, you're going to need to be able to explain more detail about what goes on. Okay. Now, so in a nutshell, what eutrophication is, it's where farmers chuck fertilizers on a field. The fertilizers run off the field into a river, a lake, a stream, and they make all the green stuff in the stream grow very, very fast, and it kills the stream. Right? So what I'm going to do now is I'm just going to go through, okay, eutrophication in a little bit more detail. So, number one, nutrients run off the land into the water. Okay, so that's the first stage. Number two, aquatic plants flourish and they grow rapidly. All right, so now what you can think now is for anybody doing higher, right, then this is probably a decent question that could come up because foundation don't need to know much about it. Right, so when we're talking about aquatic plants, we're talking about the plants at the bottom of the sea, bottom of the lake. Number three, the algae that's on the top also grow rapidly. So what you can do on the top of there, you can see algae, they're those kind of green dots, okay? And what you then do is you get, number four, at the top right here, you get an algal bloom, right? So if you ever go past a pond of any sort and you see kind of the whole surface of it green, right, that's an algal bloom, okay? Then, if you think about it, if you've got green covering the whole surface, right, sun can't get through. So therefore, if sun can't get through, aquatic plants die. Once the plants underneath the surface die, they start to decompose. And it is microorganisms that cause the decompose. And they do that via respiration. Okay, so if you think about it now, the aquatic plants have died. So therefore, they start to rot, which means they're not putting oxygen back into the water. And because the microorganisms are doing respiration, the oxygen levels decrease massively. And eventually, the whole water becomes anoxic. Anoxic means no oxygen. That means nothing can survive. Okay, fabulous 6-mark potential question. Right? Nutrients run off into the water. Aquatic plants grow, they do really well. Algae grows, algal bloom on the surface. The algae prevents sunlight going in, so therefore the aquatic plants die. When the aquatic plants die, they then do aerobic respiration and they take more oxygen out of the water. Eventually, the water becomes anoxic, which means no oxygen's in there at all, which means nothing can live. Okay, that's eutrophication. Right, variation. Okay, so we're kind of sort of another decent sized topic. Okay, so variation, right? You are all different, okay, and you are all different because of two main things: genetics and the environment. Okay, so Hitler is very, very famous for his experiments that he used to do. Okay, and one of his favorites was getting identical twins. Okay, and he used to get identical twins, right? And he used to do loads of operations on to see how similar they were. But then he also used to get identical twins and put them in different places in the world. So he'd send some to Siberia, a work camp, right? And then he might keep some in the center of Germany, of Berlin, right? Where they might live nice and happy. Okay, and his experiment was to see if they were going to get any different. Okay, so their genes are the same, so therefore should look the same. However, one in Siberia working in a work camp is going to be pale, probably very skinny, but quite muscular because they're doing a lot of hard work. Okay, the one that is then in the middle of Berlin, right, is probably going to be more tanned because the weather's better. Okay, they're probably going to be fatter because they've got better food and less healthy. Okay, so basically, variation is a combination of genes and the environment. The variation is caused by genes and the environment. Okay, if you go to Brazil on holiday, you're going to come back brown, right? If you go to Torquay on holiday, right, you're not going to come back brown. Okay, variation. I've just put an example of a family tree on there, right, just to kind of give you a little bit of an idea about what it looked like. Okay, so again, there's a key. There's a key at the bottom of the page there. Okay, where the red square is an affected male, affected female. It could be any disease ever, right? I couldn't find a proper GCSE question based on this. Okay, but again, you just need to be aware that something like this could happen. So what they'll do is they'll give you, these are then kind of the main parents at the top here. Dad is affected. Okay, it could be a disease, it could be arthritis, it could be Alzheimer's, it could be, anything. And what you then have to do is they might ask you about the route that the disease is then going to take. So what you can do is you can see dad has then had a boy with the same disease, a daughter with the same disease, and another daughter with the same disease. Right? Maybe they're asking about sex linked. Okay? Is that a sex linked disease? No, because two daughters have also got it. Okay? Right. Extinction. Okay, so definition of extinction is where no members of a species survive. Okay, right. And what I'm going to do is I'm going to show you the picture in a minute where you can again think about the main ones that you sort of would talk about in extinction. Right. Now, before we mention about the reasons, just remember about what a species is. Okay, so a species is, let's say it's an animal, but it cannot, a species cannot reproduce with another species. Okay, so an individual species cannot sexually reproduce with another species. Okay, or have, no, they can reproduce, but they can't have fertile offspring. Okay, I'll just change that again. So what you've got is you could have a tiger and a lion that are different species, but what they can do is they can replicate, right, or reproduce, and they can have a liger, which is a lion and a tiger, but the liger is infertile. So a species can, in some circumstances, replicate or reproduce, but it cannot have fertile young. Another good example is a horse and a donkey. Okay, so a horse and a donkey could do jiggy jiggy. Okay, and what they would then do is they could then produce a mule, right? And what a mule is, it looks a bit like a horse and a donkey, but mules are infertile. Okay, so that's a species. Right, now, causes of extinction. Now, what you need to do now is, in a way, you kind of need to think, right, these are my four favorites. Okay, so we've got things like human predation, right? That is a pretty good one to do with making extinctions. We've got loss of habitat. We've got invasive species. Think about the squirrel. Okay, because the squirrel, the grey squirrel rather, is an invasive species. It's come in, right, and then what it's done, it's then taken over from the red squirrel, and it is an invasive species. That could also be better adapted competition. The grey squirrel is better adapted because it's bigger and stronger. And it's also more aggressive. Climate change is also another cause for extinction, right? Eventually, there's going to be all sorts of problems like penguins and polar bears and stuff like that are going to suffer. And pollution. Okay. Now, the big extinction ones are your asteroid strikes. Okay. For like when the dinosaurs went. Okay. Now, again, the best example is the dodo. Okay. You probably have talked about the dodo in school. All right. I think it was about maybe 150 years ago, there was dodos, right? And basically what the dodo was, it was a big turkey, right? Probably about maybe 3 foot tall, massive, great big beak, can't fly, huge, great big fat thing. Okay. The dodos, before man arrived, had no predators. There was nothing on the island. Was it Madagascar? There was nothing on the island, right, that could hurt a dodo. Men arrived, they hunted, but the main things that actually caused the problems with the dodo was rats ate the eggs, dogs chased and killed the dodos. And eventually what happened is, because they were a bit useless, they were all then killed. Okay, that is then extinction, where there is none of the species left at all. Okay. Right. Again, I'm just flying through topics here because again, it's very difficult to understand or predict what's going to be going on. Right. Antibiotic resistant bacteria. Okay. Right. Now, before I kind of start talking about those diagrams, right, there is really 3 levels of bacteria, of antibiotic. The first one is the one that you would then mostly get from the doctors. Right. You know, you go to the doctors and you got a bit of a sore throat or whatever it is, and they give you an antibiotic. They give you a general antibiotic that in most circumstances it'll kind of do its job, but it's a bit rubbish. Okay. There's been huge amounts of exposure to bacteria for that. Number 2 is pretty much one that is mostly used in hospitals. Okay, so these are kind of very generic kind of titles. They're not official scientific titles. It's just to give you a bit of an idea. The third one is kind of like for major operations that they literally do not use very much. Okay, and the main one is called methicillin. Okay, methicillin. Okay, that is the main strongest antibiotic that hospitals have. So methicillin. Okay, now antibiotic resistance bacteria. Okay, what you've got to do now is you've got to think about the process that happens. All right, so all over you, what you've got is you've got bacteria. Okay, you take a course of antibiotics, those antibiotics will pretty much most of the time kill all the bacteria. If they don't, and I'm kind of looking at my second circle here, if they don't kill all of those bacteria, that little blue one in the middle there is then immune to it. Okay, it's resistant to that antibiotic. Okay, that one then, what it can then do is it can then reproduce. And then what you've got is you've got tens of billions of those ones that are now immune to that antibiotic. And now you can't treat it with that antibiotic. So currently there is lots and lots of kind of bacteria that are immune to our kind of level 1 antibiotic. Then that antibiotic or that bacteria that is resistant to the antibiotic, it will then replicate. And eventually what will happen is it gets given to people over and over again. And maybe that one there becomes resistant as well. And as soon as that one there becomes resistant to the next level of antibiotics, we've now lost our second family of antibiotics, leaving only the strongest ones left. Okay? So basically what you've got to do is you've got to make sure, or doctors make sure, that the least exposure that there possibly is for antibiotics to bacteria, the better. Okay? And what we have to do, or what doctors have to do, is they've got to protect stronger antibiotics that there is. Right? Now, there are two main kind of bacterial infections that are immune to every single antibiotic that we've got. The first one, the one that they are going to mention, is MRSA. Okay. My auntie, she was about 80 odd, right? And she had cancer, right? She went in for her regular treatment for her cancer. While she was in hospital, she caught MRSA, right, in hospital, and then she never left again. Okay, so it wasn't the cancer that killed her, it was actually the MRSA in hospital that killed her. Okay, and MRSA can be found more in hospitals because it is such a sterile kind of environment. MRSA stands for methicillin, which is the name of the actual antibiotic itself, resistant Staphylococcus aureus. Now, you don't need to remember the kind of the big posh name for it. You just got to remember that MRSA is immune or resistant to every antibiotic that humans have. Now, the other one that I'll be very surprised if you're actually asked about is called supergonorrhea. Okay, so supergonorrhea is gonorrhea that has then mutated to be immune to all of those antibiotics. If you ever catch supergonorrhea, right, you're in a bit of trouble. Okay, right. Classification. Okay, now this is again, it's a question until about 2 or 3 years ago never came up. Okay, but in the last 2 or 3 years, it's come up every single year. All right. Now, every single animal is classified. Okay, and what they do is they'll do it into a certain kind of structure. So in this column here, right, those black words, that is us. Okay, so we are in the kingdom animals. Chordata means has a backbone, has a spine. Mammals means you are hairy and you produce live young, right, and suckle. Primates, opposite thumbs. Hominidae, stand up. Homo sapien is then our genus and our species. Okay, so that, you don't need to remember that. That is our kind of classification. Okay, so what they could do is they could look at us, right, an alien, they could come in and they could look up this and they could work out that we were Homo sapiens, humans. On the right-hand side here, right, then I got coyote and I got grey wolf. Okay, now they are similar, right? But again, I've just put that on there just to kind of give you an idea. Now, what you need to be able to remember on this one, okay? You need to remember that order, the names of it, and that correct order. Okay. Now, the last two kind of exam questions that came up were they kind of missed out boxes. So they kind of missed out that box and they kind of missed out maybe a box over here. Right? They gave you the information to then be able to try and fill in the table. Okay. And the bit that every single person that I know got wrong and from loads and loads of different schools is people didn't know this order here. Okay. So at the top, right, what I've then got is I've got a little mnemonic to try and remember it. King Philip came over for green soup. Right, now you can change the wording if you want. King Philip came over for green soup. So it generally wouldn't surprise me if they don't do another table or something about classification that you've then got to remember that order in orange. Okay, so king, kingdom, Philip, phylum, class, order, family, genus, species. Okay, generally, I would remember that. Okay, also, don't forget, this PowerPoint is going to be on, well, it is already on Google Classroom. Okay, so King Philip came over for green soup, right? Whatever, okay. So I was just thinking about one that I did for when I did my A levels, and it was not very appropriate to say online, probably. Okay, right, classification. There's a guy, Carl Woos, Woesi, right? And what he did is he came up with the names of the kingdoms. All right. Um, not much about him really. Okay. Now, evolution. Okay. So evolution is the biggie, right? Charles Darwin. Okay. You've obviously all heard of Charles Darwin. Okay. And what he did is he came up with the theory of evolution by natural selection, survival of the fittest. Okay. So all of the kind of the best adapted animals are the ones that survived. His theory all sparked from when he went to the Galapagos Islands. Okay, they're somewhere off the west coast of Argentina, right? Many thousands of miles from pretty much anywhere. And what originally there was an island, and on that island they had a finch. So a finch, just a small bird, a bit like a robin or a kind of a small starling or something like that we have. Okay. And his theory was that on that original island, there was just one type of finch. Over many hundreds of years, probably thousands of years, what happened was the islands separated. Okay. Just literally by kind of tectonic movement or could be volcanic activity or something. Then on each one of those individual islands, what there was then was a different finch. Okay. But what they all were is they were all finches, but they were then different species. Okay. And this is then when his theory of evolution came. Right. So if you look at kind of this one that's got a big head at the top there, that is then adapted because probably on that island there was those nuts. So the bird's then got a big beak. What it can do, it then crush nuts. This one on the bottom right hand side here, right, it's got quite a thin sort of sharp beak. That might be for insects. Okay. This one over here would then have had a different food source on that island. So Darwin, his theory of evolution was based on the fact that the most or best adapted animal is then the one that survives. Okay, survival of the fittest. Okay. Now the issues were that what with it was he came up with a book in 1859. Okay. At that time, there was really only kind of one theory that was out there, which was your creation theory. Nobody, right, thought anything different other than the fact that God created everything. Right? So, number one, the reason why it wasn't accepted straight away was because God obviously created everything, creation theory. Number two, there was very little evidence, right? Because all Darwin's come up with, he said, oh, there's a load of birds on these islands here. And also, number three is people didn't know how inheritance was actually done. Okay, so people didn't understand about sperms and eggs and all those kind of things. Now, on the syllabus, okay, and he's never ever come up. He came up on a separate swan about 2 years ago, I think. There's also another guy called Alfred Wallace that nobody's ever heard of. He was around at exactly the same time as Charles Darwin, and he came up with exactly the same theory. Okay? So it would not surprise me if they start talking about Charles Darwin and his theory of evolution. Alfred Wallace might be mentioned, and he came up with exactly the same evolution theory, but they never ever met. It was only after they'd both died that they realized that they came up with the same theory. Okay, so he could come up, but more likely it's why did Darwin's theory not get accepted? Number one, creation theory, God. Number two, no evidence. Number three, they didn't know how inheritance actually happened. Okay, right. Now, there was another guy, right, who also came up with another theory. Okay, I kind of like this one. All right, and his name was Lamarck. Right, and his theory was then Lamarckism. Okay, so if you think about Darwin, so I'll just do Darwin on this right hand side here first. Basically, if you've got two giraffes, one giraffe's got a long neck, one giraffe's got a shorter neck. Which one's going to survive? The one that's adapted the best, which is the one with the long neck. So the giraffe that's got the longest neck, this one here, is going to survive because it can eat the food more. So therefore, that'll be stronger, that'll be fitter. That's the one that's going to have the babies. Okay, so that is then the theory of evolution. Now, Lamarck's idea was great in the fact that he thought that if there was a giraffe and it kept on stretching its neck to reach leaves in a tree, its neck would get longer. Okay, and then if it kept on stretching, then it thought its neck is going to get longer and longer and longer and longer. Lamarck's theory then was the fact that that giraffe with the stretched neck is then going to have babies with a longer neck. Right? So he thought that characteristics could be passed on within one generation. Okay? So that's a bit like me going to the gym and being like massive and muscular, right? Having a kid, and that kid would then pop out massive and muscular. Okay, which might sound stupid, okay, which I guess it is, right? But that was Lamarckism. Right, back to a little bit of kind of environmental stuff. Okay, is a couple of definitions, right? We've got the environment, right, which is the conditions where an organism lives. The habitat is the place where an organism lives. Okay, so your habitat is at this moment in time probably your bedroom. Okay, that's where a organism lives. Right, one of the problems with us and extinction is knocking down the habitats. So if I burnt down your bedrooms, right, you would all suffer because your habitat is gone. A population is the individual versus species. Okay, so you are all humans, you are all population, and you are species living in the UK. A community is all sorts of different species, right? So we all live as a population of individual species, but our community is the birds and the mice and whatever else out there. Right. Competition. Okay, so what we've got then is we've got competition is where we have plants competing for light, space, water, relatively straightforward. Animals competing for food, mates, territory. Okay, that's competition. Everybody's competing. You're competing against the other 100,000 year 11s doing their GCSEs. Okay. Interdependence is not as obvious. Interdependence is then when you have two species that need each other. Okay, so the example I always think of then is like a rhino, and I never remember the name of the bird. There's a little bird that kind of sits on the back of a rhino. Okay, so the rhino has loads of ticks and diseases, right, that could eventually make it very ill. The bird eats those ticks and diseases. So therefore, what they are is they are interdependent. They need each other. So the rhino does well out of it because the bird eats all the ticks and the infections. The bird does well because it's got loads of food. Okay, so that is an interdependence where they both need each other. Right, adaptions. Okay, this is actually going straight the way back to probably key stage 2. Okay, but obviously it's a little bit more detail. Right, so we've got 3 types of animal adaptions, and we'll talk a little bit about plants as well. Right, we've got structural, okay, which is what they look like. We've got physiological, and we've got behavioral. Okay, so the most likely sort of questions they're going to ask is they're going to give you an animal, right, and they're going to ask you about how it's adapted and why. Okay, so polar bear, right? If you think about the way it's adapted, so if you look at structural, it's white. So a polar bear is adapted by being white, so it can then be camouflage. That's structural. Physiological, right? Sort of parts of his body that might save its life is the fact that it's going to have very, very thick fur. Okay, and I think it's even got some like 2 layers of fur, and the actual hairs themselves are hollow. And it also has a very, very thick layer of fat. So physiologically, a polar bear has thick layer of fat, so it can control the amount of heat loss. Behavioral, a polar bear hibernates. So therefore, what it does is for, I don't know how many months of the year it is, 5 months of the year, 6 months of the year, it goes and sleeps in a little snow cave somewhere, reduces its heart rate, reduces its body temperature, and then it hibernates. Okay. Now, they are kind of 3 different sort of animal adaptations. And what you've got to do then is you've got to look out for what they're actually asking. Are they asking for what it looks like? Right? Are they asking physiologically about how it's different? Or are they asking about the behavioral? And what they'll do in that is they'll give you the question, right? And then you've got to pick out which one they are looking for. So just be a little bit careful on that one. So if it's structural, kind of what it looks like, you're talking about it being white. If it's physiological, it could be the layers of fat. It could be the large feet for spreading the area across the top of the snow. If it's behavioral, it could be hibernation, where it goes to sleep for 5, 6 months of the year. Right? So just have a little look about what they're actually asking for. If it's structure, physical kind of part of it, or its behavior. Okay. Now, again, they could ask you a plant, right? And the main things with a plant, right, is, I'll just use the example of a cactus, right? We've talked about in biology paper 1, okay? We've talked about in biology paper 1 about the structure of a leaf, okay? And in a cactus, the biggest adaptation of a adaptions of a cactus are the spikes to prevent animals eating it. And it's also got a very, very thick, waxy layer. That's not to prevent water getting in, remember. That is then to prevent water getting out. Okay. Again, there's so many individual little topics, and basically what I've done here is I've just sort of almost like put individual topics onto each slide, right? Okay, extremophiles. Now, again, you'll have talked about this at school. Now, whatever they're going to sort of ask you, right, you've always got to remember that a file is something that is liked. Okay, so you can always talk about the paedophile joke, right, where obviously file means like, ped means children. Okay, now obviously they're not going to talk about paedophiles in your exam, right, but what they are going to do is they could mention another file. Right, and what I've put on the board on the screen there is 6 possible examples. A thermophile
likes high temperature conditions. Again, the words in there, right, are going to tell you kind of an answer. A halophile likes the halogens. Salt, sodium chloride. Acidophile, alkaliphile, can't even say that one, right? They like acidic conditions, alkaline conditions. Psychrophile, low temperatures. Radioresistance phile, high doses of radiation. Okay. Now, again, I'm never ever going to predict what extremophile they're going to talk about, but they will have the name phile in them. And you've then just got to look at the little word that goes with it and just try and have a little think about what is it they like. Okay. Right. Bit more kind of environment stuff. Predator-prey cycle, right? I suspect they're not going to really ask very much on this one. Okay, but if you kind of look at it, it's kind of a bit of a logic thing, right? You've got a little cute bunny rabbit, population goes up. Okay, if the population of a little cute bunny rabbit goes up, the population of its predator goes up. When the predator then starts eating all the bunny rabbits, the population goes down. Then the prey population goes down. Okay, that's a predator-prey cycle. Okay, I don't think they're going to ask much on that. Okay, I'm more worried about or bothered about the fact that they're calling it a predator-prey cycle. Right. More environment kind of stuff. Okay, right. So we've now got the greenhouse effect, global warming, and climate change. Okay. They could talk about what causes global warming. They could talk about what causes climate change, right? But I think the most important thing on all this is remembering that the greenhouse effect causes global warming, which causes climate change. Okay? What they all do is they all kind of fit in together, right, as a process. So the greenhouse effect, and we'll talk about that in a second, causes the globe to warm up, which then causes climate change. Okay, so remember that order. Right. Greenhouse effect. Okay, if you do geography, you'd have done quite a lot on this. Okay, where what you've got is then you've got the sun, right? The sun's rays enter through the Earth's atmosphere, and they can do that quite easily. Then, when they enter into the Earth's atmosphere, okay, what they are is they will then turn into it's like a thermal energy, and they will hit the Earth's atmosphere, or hit the Earth rather, and then they'll try and rise up. Okay, so what will happen is sun's rays come in, they heat up the Earth. Then what you've got thermal energy. That thermal energy then tries to rise up. Okay, for about the last, I don't know, 10,000, 50,000, 100,000 years, okay, it's been kind of status quo where it's all been kind of the same. What we have now done is we have now introduced mainly carbon dioxide, methane, water vapor, and sometimes they sometimes call it the nitrogen gases. Okay, but the main two are those at the top there. We have then introduced carbon dioxide and methane into the atmosphere. Okay, and by doing that, we have then thickened the greenhouse layer, okay, the greenhouse gases. So the logical thing then is that sunlight comes in, historically, the same amount of heat may well then go out. Because we've now made that layer thicker, when the sunlight comes in, less heat gets out. And then you go to my little diagram here, the greenhouse effect, because we've then thickened the layer of carbon dioxide and methane, that greenhouse effect causes the globe to warm up. Okay. Now, where does the carbon dioxide and the methane come from? Carbon dioxide is from your combustion. Okay, so from burning fossil fuels, from deforestation, from car engines, anything that then is producing carbon dioxide. Methane, normally talk about it from two different places. First one is cattle, mainly cows. Okay, so what cows are doing is they're farting and they're burping. Okay, and from both ends, they are then producing methane. And the other place for methane is paddy fields, right? For some reason, rice, when it's actually growing, produces methane. And if you think about the amount of rice that is actually made in the world, right, for billions of people to eat, there's a lot of rice that is then producing methane. They are then kind of the two main gases and kind of where they come from. Okay. Now, global warming. Now, what I've done on here is I kind of sort of got a diagram that kind of sort of summarizes the whole kind of global warming kind of process. So starting here, global warming is kind of the main thing. The global warming is caused by the greenhouse gases. Right? Two of the big things that cause the greenhouse gases are deforestation and burning of fossil fuels. Okay, so global warming is caused by the greenhouse. And two of the biggies is deforestation. If we're then chopping all the trees down, there's nothing to absorb all the carbon dioxide. And we're also burning all the fossil fuels. Global warming, the effects of it, climate change. Okay, so climate change, the biggest issues that there are then is more droughts, rising sea levels. Okay, because if you think about it, wherever you might live in the world, okay, I live in Lytham, right, which is literally, I know, maybe I live about what 400, 500 meters from the sea. Okay, so if the sea levels rose probably about 3 or 4 meters, right, my house would then be underwater. Okay, so climate change, increased droughts, rising sea levels, more extreme weather. Certain places are going to get hotter, certain places are going to get colder, right? They actually talk about the UK is actually going to get a lot colder, even though climate change is happening. What are the solutions? Okay, the solutions are plant more trees, save energy, switch to better energy resources. Okay, so what I've tried to do on this little diagram here is global warming, number one, what are the causes? Global warming, what are the effects of global warming? Number three, what are the solutions? Plant more trees. It was on the news about a week ago that the Chinese are planting about 1 billion more trees a year. Save energy, right? Do you turn your little red dot off on your TV when you go to bed at night? Okay. Do you turn everything off when you go to bed at night? Switch to more energy efficient resources. So, like Octopus Energy only provides electricity from renewable energy sources. So, wind power, tides. And then number four, why don't we do it? First one, right, we can think about Donald Trump in every single one of these now, right? So just get a picture of Donald Trump in your head, okay? Lack of awareness, right? People aren't aware of what they're doing that's causing the problem. Okay, so maybe somebody way out in the middle of, I don't know, Africa or whatever it is, and they're sitting there burning logs or whatever it is, they're not going to be aware of the fact that what they're doing is producing carbon dioxide. Okay, now they've probably got no choice, okay, because most of it is from the Western countries. It costs money. Okay, so we don't change our habits because it's too expensive. Okay, and then we've got expensive there as well. Okay, because if you think we've got enough electricity produced now by our coal oil power stations, why are we going to build something else and take those away when it's already working? Okay, so if they start talking about global warming, don't forget greenhouse causes global warming. Global warming causes climate change. If they talk about global warming, I've then given you 4 titles to talk about. What are the causes of global warming? What are the effects of global warming? What are the solutions to global warming? And why don't we do it? So why don't we do it is people are unaware or it's money. Okay, right. Decay. God, there's so many different topics in this. It's just a nightmare. All right. Decay. So decay is all about temperature, water, and oxygen, right? If you've got a dog, right, and you take your dog for a walk and it has a little poop somewhere, okay, then that little dog's poop, right, to actually disappear needs to be of the correct temperature. It needs to have water and it needs to have oxygen. And basically, that's for the microbes to work, right? So if your little dog's poop is too cold and it freezes, it isn't going to decay. If it dries out because it's sunny, there's no water, it won't decay. Not quite sure how you get rid of the oxygen. Right, carbon cycle. Now, again, carbon cycle in some form comes up every year. Okay. Now, what I've got on there is I've got kind of a standard kind of carbon cycle diagram. Okay. Now, what I think the best way of actually kind of addressing this is there's no point in learning the carbon cycle, right? Because they will put a different one on your exam paper to the one that you found and learned. These first 5 things, okay, are the main things that they're going to ask about with the carbon cycle. Okay. And often what they'll do is they'll draw one and ask you to label it, or they'll draw something and they'll ask you to explain how the carbon cycle functions, how it works. Okay. So the carbon cycle is all about photosynthesis. Okay. And you can see that over on this left-hand side here, there's a tree photosynthesizing and it's taking carbon dioxide out of the atmosphere. The second thing is respiration. So basically everything that isn't a plant is respiring. So we've got Flossie the sheep in the middle there. Okay, Flossie the sheep is respiring and it's putting carbon dioxide into the atmosphere. It then poos and it's got its waste products. The waste products are then eaten or used up by microorganisms. They die, and when they die, they rot, which is then respiration again. Number 3, combustion. The main place for combustion is in a factory that is then pumping out carbon dioxide into the atmosphere. The carbon cycle, there's 3 of the main words. Death is a possibility, but that's respiration again. Decay is then when things then rot. Okay, and we just talked about decay then, but it is all about microorganisms. Okay, so I'm just going to remind you now, right? If they talk about the carbon cycle, they are wanting you to write down something to do with photosynthesis, respiration, combustion, and possibly decay. Okay, they are the kind of the fundamentals for the carbon cycle. Right now, very, very quickly, right? Just while we're talking about the carbon cycle, right? We need to think about where carbon dioxide is stored. Okay, now I don't think my next slide allows me to do that. That's the water cycle. Okay, right. So let me just clear those there. Okay, and right, carbon dioxide. Where is carbon dioxide stored and that we are messing up? Firstly, carbon dioxide is stored in trees. Okay, now that's not the biggest one, right? And again, you've got to remember that if we're chopping all the trees down, right, then what we're doing is we're then preventing carbon dioxide going into trees. The biggest one is in water, particularly the sea. Vast majority of the carbon dioxide on planet Earth is dissolved in the sea. Where else is carbon dioxide stored? Fossil fuels or coal and oil. Okay, so over many, many millions of years, carbon dioxide has gone out of the atmosphere and it's then stored in coal and oil. And what are we doing? Burning it. So that's the best one, water. Fossil fuels is then the next best store. Number 3 is then sedimentary rocks. Okay, so sedimentary rocks are quite soft rocks, right? And originally what they were like shells of animals. Those shells of animals have then been crushed over time, okay, and then eventually what's happened is they turn into sedimentary rocks, but they are then a source of carbon dioxide. Number 4, trees. Okay, so that's a carbon cycle. Water cycle, kind of a lot more kind of simple, right? So what you'll do is you'll start with water on the ground. Okay, it then evaporates, condensates, which is basically turning into clouds. There's also transpiration from plants, condensation, so the clouds precipitate. It then goes down the side of the mountains, surface runoff, back into the water again. Okay, so that is the water cycle. And again, you just got to use these words. You've got to use evaporation, condensation, transpiration, precipitation. Okay, that's the water cycle. Now there is other one word, right? I quite like that diagram. Now the other word, and one of the teachers at my school is obsessed with the fact that this is going to come up. It's called percolation. Okay. Now percolation is where water lands on a surface and it kind of goes into the surface and it goes in between the molecules. Okay, so that is percolation. If you like a cup of tea, what you do is you put your tea bag into tea, hot water, and the water goes through the tea leaves. That's called percolation. Okay. Right. Abiotic and biotic factors. Okay. So abiotic is non-living. Biotic is living. Okay. So if they start talking about abiotic and biotic, right? The non-living, things like light intensity, again, make sure you use, that's supposed to be light intensity, not quite sure what it does that. Temperature, moisture levels, anything that's kind of non-living. Biotic, availability of food, right? These are then the things that affect where something lives, new predators, pathogens. Okay. We've just kind of talked about that one, which is factors affecting. Again, and it's another little kind of factual thing that they seem to like asking about is how do humans use land? Okay, so humans use land to basically take it away from every other living thing. We use it as building and quarrying, farming, dumping waste, and then we destroy the peat bogs. Waste management, we've kind of talked about that one, right? Genetically modified crops. Okay, these are then crops that have genes from other organisms. Okay, so if you see anything about genetically modified, it's not selective breeding. So selective breeding is where you then get a characteristic, and then what you do is you breed that characteristic into the next one. So, for example, I used to have a pug, right? And the pug, I know 500, 300 years ago, had a long nose. Selective breeding is then when a pug was then born with a shorter nose, then bred again and bred again and bred again until you ended up getting the pug that you then look like. If you like Alsatians, Alsatians used to have a straight back. Now Alsatians, what they've got is they've got a curved back and they've got low hips. That is selective breeding. Genetically modified things are stuff like where a potato has disease resistance put into it. Animals can produce more milk, right? There's a certain type of cow, right, that naturally has a massive, great big bum, right? And what they use that for is they use that for certain steaks. Genetically modified dogs, large unusual flowers. Okay, they're still trying to genetically modify roses to make a perfect black rose, but nobody's ever made it yet. Fossils. Okay, so what a fossil is the remains of an ancient organism. Okay, and there is three main types of fossils. Okay, fossil number one is where something has not decayed. Your woolly mammoth found in Siberia. So what they'll do is they're in the permafrost, they're frozen for many hundreds of years, pull them out and they're still hairy. That is the first type of fossil. Part number two is kind of the one that you're more aware of, where you might have a shell, right? And you find it in a rock. That's then where the shell has been replaced by minerals. And fossil number three is there could be a habitat. So somewhere in Scotland, there is dinosaur footprints where a dinosaur has stood in mud and that mud has then turned into a fossil. Okay, right now, I think that this final then little bit is probably the only higher section that is then to do with genetic engineering. Okay. So what I'll do is I'll literally just go through this, right, for a couple of minutes. Okay. But before anybody goes, right, because we've gone over the 2 hours already, is have a look at the PowerPoint. And at the end of the PowerPoint, what there is there's then 2 quadratic kind of style questions. Okay. Now I've tried to make them a little bit different, right? So the first one is about a circular field. So it's kind of talking a bit of maths. Okay. And there's answers following it. And then the second one is then about an L-shaped field. Okay. So again, just have a little look through, right? And just make sure that you are aware of how to actually estimate the number of plants in a certain area. Okay. Right. And also, you've got to know the quadrats one. So I've put then quadrat questions up there, right? But there's only 2 required practicals and combined: reaction time and quadrats and transects. Quadrats and transects, it wouldn't surprise me if you don't get a quadrat question and a transect question. So please, 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 right, after you finish here, look at those methods again, right, and make sure that you can then say a method from start to finish where you are actually explaining in your end bit exactly how you will then tell an estimate of the number of daisies, flowers, whatever it is in a field. Okay, because it will be on. Okay, right, and then reaction time, okay, with the ruler. Okay, right. So again, think about the question, read the question, and make sure whatever it says you answer. Okay, don't make anything up. Right, go on my website, not my website now, on Google Classroom, get the PowerPoint, go through it again. Right, and any little bits that you don't understand, right, research it, because that covers every single little bit of the syllabus. Okay. At about 8:15, if you do have any specific questions, okay, probably about 10 past 8:15, log back on, right, and I'll try and answer those specific questions if you have anything. Or you can email me, right, and then I will then try and email you back, okay, because it's kind of quite important for you guys at the moment. All right. So anybody that is foundation, right, good luck, right, you can log off. That's said nicely.
Thank you.
Bye. Thank you.
Cheers. Okay, now, higher wise, right, there is literally only this kind of section that is specifically higher on the combined paper, okay, which is our genetic engineering, right? Now, what I've got on here then is I've got kind of the process for it, okay? So what you have is you have this kind of pink bit down in this bottom sort of section here is your plasmid, right? So if you remember a bacteria, right? So what a bacteria has, it doesn't have a nucleus, it has a little ring of DNA called a plasmid. Okay. And what they do is they use a restriction enzyme. Okay. And with that restriction enzyme, they cut out a small chunk of the plasmid. Okay. Then using another restriction enzyme, what they'll then do is they will then put another end in or another piece of DNA in there. Okay. To fix it in place, they then use something called DNA ligase, however you want to pronounce it, I don't care. Okay. That is then called recombinant plasmid. Okay. So you've got a ring of DNA from a bacteria. You use a restriction enzyme to cut out a chunk. You use another restriction enzyme to put in a chunk. You fix it with DNA ligase. That is then called recombinant plasmid. You then put it back into a bacterial cell. Okay. Now, the most common one that is ever used for that one is insulin. So what they'll do is they will get the plasmid. They will cut out a certain chunk of the plasmid's DNA. And they will insert this kind of yellow bit here. They will insert the genes from a human that says, make insulin. Then they fix it in place with DNA ligase. That is then called recombinant plasmid. You then put it back into a bacteria. What can that bacteria now do? That bacteria can now produce insulin. And what they do is they grow billions upon billions upon billions of them. That produces the insulin, and then you can then filter the insulin out. All right. Now, again, genetic engineering, that's kind of the way that it works, right? And I can't obviously predict which sort of genetic engineering kind of genes they're going to talk about, okay? But it's all to do with having a vector, which is usually bacterial plasmid. And what they do is they insert the genes in, okay? So there's kind of other examples of like axolotls, okay? So axolotl, right, is kind of, it's like an underwater, looks a bit like a lizard that never really goes through maturity. Okay, and what they can do with that is because it has incredible kind of regeneration powers, right? They can chop legs off and it can grow legs, right? But also what they can do is they can change the genes in them, right? And they've got axolotls now that have got 2 arms that glow fluorescent yellow because they've taken a fluorescent or fluorescing gene from one creature and they've added it to another creature. Right, now, risks and benefits. Okay, this is then the final kind of combined slide, is risks and benefits of genetic engineering. Okay, the risks, what benefits one plant might harm another. Ethics, is it right to play God? The genetically modified plants are often more expensive. So if you go to a garden centre, you can buy an old traditional rose, and then you can get a genetically modified rose that is then like immune to black spot and other things like that. And the new plant is more expensive because it's gone through processes. Can cause allergies because we don't know about it. The pollen that's produced could go anywhere. So if you've got a field of genetically modified corn, it could blow onto another field. Okay. Benefits, it's a lot faster. You can get increased crop yields. You can have herbicides actually introduced into the plant. And you can also have the plant that can produce toxins to prevent it actually, to make it actually produce a poison in it to discourage insects from eating it. So genetically modified, okay, main benefits or main risks is not ethics, right? We don't know the full implications of everything. The main benefits is you get better crop yield, and you can also have them producing toxins so that insects don't eat them. And you can also have herbicide resistance. It's also faster. Okay. Right. Now, again, right, for those people that are going to disappear off, right, because it's going to be separate science stuff only in a second, is have a go at these 2 questions. Okay. I've just made them a bit weird, a circular one and an L-shaped one. Okay. Right. So have a quick look. The answers are there, but try and work them out. Okay. Right. So for anybody now that is combined, right, good luck. Don't forget, answer the question that is sat in front of you. Okay.
Thank you.
All right. Cheers.
Thank you.
Thank you.
Right. Camera's on then. Don't be shy. Come on, Zishan, I want to see that beautiful face. There we go. Daniel, go on to any of the girls that are shy. Okay, so now obviously what I'm going to do now is we can kind of just open it up, because obviously when there's about 15, 20 people on there, okay, it's hard for me to then ask people. Now there's quite a lot of extra separate science stuff that then we kind of need to go through. Okay, and what I will do is I will kind of throw it back at you sometimes. Okay, but generally, right, we just gonna kind of get through it. Okay, because I've got about 56 different slides, right? Because the problem with biology is there's a hell of a lot of facts, right? Because in that first 2 hours, every single slide was a different topic. Massive. Okay. Right, so we'll start off with plant hormones. Yeah, again, people can just interrupt if you just want to interrupt. Right, but what we'll do is we'll just go through the three main ones. Okay. Right, so first one, auxin. All right, how are we going to pronounce it? Okay, so what it does, it controls cell elongation. Okay, so it doesn't necessarily duplicate the number of cells. What it does, it controls whether they get bigger or smaller. Right? Uses, weed killers. Okay, so that's why they always say if you've kind of got a hormone-based weed killer, what you do is you spray it onto large leafed weeds, right? Because it doesn't kind of kill it by poisoning it. What an auxin, a hormone-based weed killer, right? Often what it'll do, it'll spray it on, and it causes cell elongation so much that the plant almost grows too much and kills itself. You can also use it as rooting powder, okay? And if you're then doing tissue culture. Right, gibberellin, again, however you want to pronounce it, okay, is germination, right? So what I used to do is I used to kind of at work, I used to kind of grow a lot of sort of stuff from seeds. I actually literally used to go to Aldi, right? And I used to go and get like fruits and stuff like that. And I used to get the seeds out in the middle of the fruits from Aldi, right? And I used to put gibberellins on them. I used to buy them on the internet and I used to put gibberellin on it. Okay. And what it used to do, it used to stimulate germination. So in my room at the moment, I've got all sorts of hot chilies. Okay. And I've got mango plants literally bought from Aldi. Okay. And they're growing. Okay. So gibberellins, if you put them on at the right time, it helps flowers. Okay. And it helps the fruit formation. Okay. So I would use my gibberellins to make it grow, make it put flowers on, and I would then get it to then make more fruit. And it works quite well. Okay. Ethene, right, is fruit ripening. Okay. Main fruit that produces ethene, bananas. Okay. So they always say don't put bananas with all your other fruit. Okay. It's very true, right? Because if you go downstairs and you put your bananas with your apples and whatever it is, bananas will naturally produce ethene more than most others. That ethene will then make all the other fruits go riper. Okay. And again, what they'll do with ethene is where they'll pick fruit in South Africa, right? And literally they'll pick fruit that is almost like inedible. They will then put it in, and when it gets to the UK, they will put ethene on it, and the ethene will then make it go ripe, and then it will look perfect like they do in the supermarkets. Okay? Again, people can just interrupt, okay, if there is anything. Right, and I am going to be asking about the plant responses required practical in a minute, okay, for somebody to tell me the kind of the method. Right, auxins again, okay, so what you got to do now is you got to think about where auxins actually kind of is, okay. So if you've then got auxins, what they are is they are produced at the tips, okay, of a shoot. Okay, they're not necessarily produced anywhere else. What they are is they are produced at the tips. Okay, light then has a massive difference or has a massive effect on the auxins. And you can see from this diagram here, right, that what happens is if you direct light, the auxins diffuse away. So the auxin diffuses away from the light, so it goes to the opposite side. And then if you remember, when we talked about what auxins do, auxins cause cell elongation. And then you can see on this third diagram here, cell elongation along this side here, those cells get longer, it's naturally going to bend over towards it. Okay, so it's the auxins that then cause that. Right? Then we've also got our kind of roots and shoots. Okay, so that there is a germinating seedling. Okay, auxins on the shoot, there's light shining down here. Okay, then the auxins will diffuse away from the light. Cell elongation will mean that the shoot will then shoot upwards. In the root, it kind of does the opposite. So the auxin goes away, right? But then it doesn't do cell elongation, right? What's the word first? Opposite of cell elongation? Can't remember what it is now. Okay, so what it does is the actual cell elongation doesn't happen, so therefore the shoot goes downwards. Okay, and then obviously you've also got gravity that is involved in it as well. Okay, right, now what we've got is we've got our plant responses required practical. Okay, and what I'm going to do in a second is I'm going to ask somebody to tell me the method. Okay, right, so plant responses required practical. Okay, right, so just spend literally 30 seconds, think about what the experiment could be. Okay, and I've got the title there written in white, which is then straight off of the syllabus, and it says, investigate the effect of light or gravity on the growth of germinating seedlings. Okay, now obviously gravity is quite hard. Okay, so I'm going to work out who I'm going to pick on in a second. Okay, so think about it. Shan's legging it. I'll pick on when you get back, Zishan. Oh, he's back now. Right, Daniel, go through it, please. Right, let me just up working again. Okay, so what I'm going to do now is I'm going to write down, okay, basically what you tell me. Okay, so unmute.
All right, so you get like a plating dish, and then you place like cotton wool on it.
Okay, petri dish, cotton wool.
Yeah, and then you add the seedling, and then you like evenly spread it out, like distribute.
How many?
Like let's just say 25.
25. So 25 seeds that go on there.
Yeah, yeah.
Yep.
And then you can place like water around it to prevent photosynthesis, like through the roots, I guess. And then
Hold on, say that again. Prevent photosynthesis of the roots.
yes, I think.
Do roots photosynthesize?
No.
They're not going to, are they? They're underneath the ground. Roots are white, aren't they? So white roots don't have chlorophyll. Okay, carry on then. Seedlings, so we've got 25 seeds rather.
Yeah. And then you place it in like different conditions. So like you put one with like next to light, you put one in like a dark cupboard with no light, and then you put one with like some light and light.
So you put one set in light conditions, okay, another one like in dark conditions, and another one in.
It's like it's got some light, but it's like not a lot, I guess.
Yeah, so you could have like direct light, and then you could have a dark room, and then you could have like sunshine coming in from a windowsill, something like that.
Yeah.
Yeah, right. So you got 3 lots of conditions. Okay, then what?
And then you like leave it for like, let's say 24 hours, and then check on it every day.
So you leave it for 24 hours, and then you check on it every day after that.
Yeah, and then do it for like 3 days or something, and then you like see the way that not sure what happens after that. I can't know.
Not sure what happens, right? Now, I know you're on the spot here a little bit, Daniel. Okay, but I'm just going to go through the variables. So, dependent variable. You haven't told me anything about any results that we're going to do. Okay, so what's our dependent variable? That is then what we're going to measure. Okay, all you've done is you've just told me, right, that you're going to have 25 seeds, right? What are we measuring? Okay, don't mess in class, right? What's our independent variable? What are we going to change? Okay, I guess you have told us that. We're going to change kind of the light, right? And then we also need to know our control variables. Okay, so what you definitely need to do is, because obviously you are going to get a quadrat question, okay, but you are also going to get either this or decay. Okay, so you need to know this one inside out. Okay, right. I'm going to ask somebody else, right? And what I want them to do then is I want them to do the method, right? And then Daniel, you need to absolutely focus on this, right? Because you've got to get every single little section of it. Okay. Bawaji, method, please. Are you there? Zara, method, please. Let's see how good Miss Matthews is.
The one that Daniel just said.
Yes, please.
Okay. So you get your like dish.
Right, we'll get a Petri dish.
Yeah. And you wet a cotton like cotton
Wool.
ball on. Yeah.
Okay.
No, you have to wet it.
I know you do. Yeah. Oh, sorry, wet. So water, wet cotton wool.
Why'd you put a question mark?
Because something came to my mind that you'd missed. Okay, keep going.
Okay, and then put 25 seedlings on there.
So 25 seedlings,
Wait, no, seeds, just seeds.
just seeds. Okay.
And then put them in 3 different conditions. So dark cupboard.
So you're saying exactly the same thing that Daniel said. Yeah.
Yeah.
Right. Okay. Right. We're missing stuff out here. Okay. Adam, start me off.
I'm not sure about the question mark, but like with the conditions you put it in, isn't like one's a dark cupboard, but then one that's like sunlight at an angle.
Yeah, but I
And
won't
then you
before
have
that. We haven't even done the first bit yet.
so you have your Petri dish.
Okay,
If
we've
you're
got
asking
Petri
someone to
dish,
say the same thing.
and then we've got wet cotton wool.
25. 25's too many, surely.
Don't really matter. You can do 25. You do a million if you wanted to count them. Right, 25 works. I normally go for 10. Okay, but again, we need to think. Zishan, are you going to save us?
All right.
All right. Get in. All right.
All right.
All
First
right.
get fixed amount of water, volume of it, so 100 centimeters cube of water.
Oh, hold on a
Put
second. There's my question mark. 100 centimeter cubed of water. Now that sounds quite a lot of water. Okay, let's just go for maybe 50 centimeter cubed of water.
all right.
Control
50 centimeters.
variable. Come on.
Yeah, control. Um, so 50 centimeters cube of water into each Petri dish with cotton wool.
Yep.
Place a minimum 10 seeds in each Petri dish.
10 seeds.
Allow time for the seeds to germinate. I forgot something. Also, put it into each petri dish in a controlled environment, so same temperature, same level of oxygen, stuff like that.
So warm place, temperature, amount of carbon dioxide, standard conditions, ideal
Yeah,
conditions.
yeah, ideal conditions.
Good.
Um, wait, I just blanked one sec. Oh yeah, place the petri dishes in one in no sunlight at all, one in partial sunlight, and one in complete sunlight.
Okay.
Then leave it in for a week.
Week. Right. Zishan, you let me down now.
Is
Okay.
it 24
Right.
hours?
No, it's not that. Right. Every single one of you, right? You need to think a little bit now. Okay. Now, Zishan, you did way the best job there. Okay.
Alright.
Right. Now, all of you, you need to think, right? What is our dependent variable? Nobody's
getting
even
to that,
mentioned
sir.
our dependent variable. Go on, Zishan.
It's the height and the direction in which it grows in.
Height of seedlings. How on earth can you possibly do a practical where you're not measuring anything? Right? All of you said then was you got seedlings, and what you're going to do is you're going to put them in certain conditions. What are you going to measure? Okay, think about it. Right, think about it a lot. Okay, right now, Zishan, that was dead good, even though you've now legged it. Okay, now what I'm going to do now is I'm going to then put up a method, and we can even add a little bit things to this. Right, and every single one of you now has to think about this. Okay, right now. Set up 3 Petri dishes containing cotton wool soaked in equal amounts of water. All right. Think about your variables, right? You've got to make sure that this is done in as much details as possible, right? So on that one there, I'm going to put 50 centimeter cubed. Put 10 mustard seeds in each dish. Put the dishes in a warm place to germinate, right? You can't assume that 25 seeds are all going to germinate. Okay. Now, what Zishan then said, he said ideal conditions. He said the same light, he said the same temperature, he said the same amount of water, which is perfect, which is that bit there. Now, once they're germinated, you've also got to have the same number in each Petri dish, right? So let's say you've got 3 Petri dishes, 10 germinate in one, 10 germinate in another, and 7 germinate in another. What are you going to do with the first 2 Petri dishes? What are you going
Take
to
3
do?
away from each one.
You take 3 away, right? Because again, you've got to think about it. Each one of those seedlings is using resources. It's using up water, it's using up carbon dioxide, it's using up light. So therefore, you've got to have the same amount in every Petri dish. Okay. Then what you do is you're going to measure the height of each seedling in millimeters. Okay. This is then what you were saying, Zishan. Okay. And what are you going to do with them? Right? You're going to measure the height of each seedling in millimeters, and you're going to record it on a table. Okay. So that is then day 1. This is then when everybody jumped to. Okay. But you would all have done not that brilliant on that because you hadn't got anything to measure. Now you put them in the conditions. One on a windowsill in full light, partial light, third one in darkness. Measure the height of each seedling every day for at least 5 days. And then you record the heights on the table. Okay, so now if they asked about the variables, don't mess in class, stay calm. What are you going to measure? You're going to measure the height of each seedling, and you've only got 7, you might have 8, right? If you want to put 25 in and 22 of them germinate, you've got to make sure you got 22. Why do you have to have the same number? Because they are all using up water. Okay, and Zishan told us that we're going to put 50 centimeter cubed of water in there. Okay, if you've got twice as many seedlings, then the water's not going to be a control. Okay, right. Now, honestly, you all need to think about that one, okay? Because I think that's the first time ever, right, that I've asked 3 people, right, and you've kind of said the same thing, right? So proper have a good think about it. Okay, right, Adam, give me 3 control variables for that practical.
The temperature of the room, the amount of water that soaked up by the cotton wool,
Right,
and
can
the
you do me a
amount
favor?
of
Can you never use the word amount either? What word are we going to use instead of amount?
volume, the volume
Good.
of water soaked up by the cotton wool, and the amount of germinated seedlings in each Petri dish.
Perfect. Okay. Right. Then we can then go through the process. Okay.
Right. So now what we've got is on my screen that I'm showing you at the moment. Okay, we've got, can you see my screen? Yep. Right. So we've got dark, we've got light, and we've got directional. Okay. Right. Now, what I'm going to do now is that is a very kind of blurry sort of table that I've then got on there. Now, obviously, what they'll do is they'll give you a set of results or however they may well do it. Okay. And what I've done on there is I've said 8 days. You don't know how many they're going to do. Okay. Well, they've done 8 seedlings rather, 5 days. Okay. You'll get a graph to draw. Okay. And then what we'll do is then there's going to be a graph and you're going to have to interpret it. Okay, so if we think about it now, we've got 3 different sets of seedlings in 3 different conditions. Okay, now the question is, which one's going to grow the fastest? Okay, Farhan, right? Out of dark, light, partial light, which one's going to grow the fastest?
Oh, light.
The light one. Okay, Daniel, do you agree? Yeah. Zishan, no, I'm not going to ask you yet. Zishan, Adam, do you agree? Yeah. Zara, do you agree?
Is it partial light?
Partial light. Okay, so we've got 3. Bawaji, are you there? Are you talking?
Yeah.
Right, which one's going to grow the fastest?
Hello.
The light one. Okay. Bawaji, just out of interest, did you get the method right before with all the control variables and stuff?
No.
No? Right, again, all right. Everybody's got to focus on that one tonight. Okay. Right, I'll show you the graph. Okay, so now my logic in my brain says the light one is going to grow the fastest. Okay, but they don't. Okay, so if I then go back to my original kind of picture here, right, those sort of seedlings that are in dark, they do grow faster. Okay, because what they're trying to do is they're trying to get to a source of light. So they literally use up all of their energy from the seed to get to a source of light. Okay, you can even try it, right? Try it with cress, right? If you get a load of cress seeds, which is basically the same as this, right? And then you try and grow them, the cress seeds will grow very, very fast in dark, but they'll be yellow. Okay, where the other ones will grow stronger, but they won't grow as fast. Okay, so the reason why they are growing faster in darkness is because they are using up all the energy in the seed to try and find a source of light. And as soon as all that energy in the seed is then gone, what will happen then is they'll die. Okay, and your partial and your full sunlight are pretty much the same, really. Okay, there's very little difference between them. Okay, because the amount of sun that's actually required is relatively small. Okay, right. So that method that you've then got there, right, you need to know. Okay, now again, if I just flick back to the very first screen, what it says here, it says in the actual syllabus, investigate the effect of light or gravity on the growth of germinating seedlings. Okay, now actually testing gravity is pretty difficult. Okay, now this device here, right, is something that actually takes away gravity. Okay, and what it is, it's called a clinostat. Okay, and what it does is it rotates very, very slowly to take away the effect of gravity. All right, so then what you can do then is you can then see the length that a shoot might grow, or the direction a shoot might grow, or the direction or speed that a root might grow without the effect of gravity. Okay, so a clinostat, right? It's literally a petri dish on its side that rotates very, very slowly. And when it's rotating, the idea of gravity disappears. Okay, right. And then what you'll then have to do, you'll have to think about your method to then sort of use a clinostat to be able to do that.
Has anybody got any questions about a clinostat?
I got a question, sir.
Go on.
Can you not also say a slow spinning motor as well?
Yeah, you could, yeah. Well, that's
Uh,
basically what a clinostat is. It just sounds posher,
Yeah.
doesn't it? Okay,
Yes.
right. Okay, right. Now, we've got another one. We've got decay required practical to come up as well. Okay, but I just thought I'd kind of break it up a little bit. Right, now, compare sexual and asexual reproduction. Right. Now, it's unlikely that that's going to come up in the combined paper, right? Because it's way more kind of the separate sort of stuff, right? So what I did is I went through it with everybody else, right? Because it could come up, right? But it's way more likely to come up on yours because it's a specific box on your syllabus. Okay, so I'm not going to go through all this again because we kind of did, right? But don't forget your howevers. Okay, so parents 1 to 2, mitosis to meiosis, because again, combined need to be aware of meiosis, okay, but they don't need to know the detail of meiosis. Okay, offspring asexual, genetically identical, right, with sexual unique. Okay, variation for asexual is only mutations. All right, and what we're going to do is we're going to go through the 4 mutations later on. The sexual reproduction, obviously, if you've got 2 gametes, that is then naturally going to cause variation. Okay. Now, again, if you get a compare question, don't forget to do your right? Slower, faster, bigger, smaller, whatever it might be. Okay, don't forget your because that's even better wording. Okay. Now, also on the syllabus, it actually talks about advantages and disadvantages. Okay, so what you've got is you've got sexual reproduction and asexual reproduction. You can use some of those other things that we've got on that other one, right? But is it necessarily an advantage that you're going to produce an offspring every 30 minutes? Okay, is that an advantage or a disadvantage? Okay. Right, so the main advantages with sexual reproduction is high genetic variability. Right, so what you got to do? You got to think about your wording. Sexual reproduction speeds up evolution. Disadvantages, it's very costly on energy. Right, so if you think about it, when a lady gets pregnant, 9 months. Right? How much energy is involved in actually doing that compared to 30 minutes of asexual? Okay. And I've got on here, usually sacrifices the fitness of one sex. Okay. So, I've never actually gone through it. Okay. But obviously, pregnancy is a pretty tough thing, right? And it can cause medical issues. Okay. So, some women can then be incontinent after it. Right? If you have a C-section, you're talking about a cut through the main muscles, the main core muscles. Asexual reproduction there's obviously no courtship. You don't have to say, do you come here often? Right? If that's your chat-up line. Okay. There is disadvantage, low genetic variation. So therefore, all those kind of bacteria are pretty much the same. And that's not particularly the best word in the world. Retards evolution. Okay, probably a better one than that is slows evolution. If you think if one bacteria becomes another bacteria, it's the same, okay, then evolution isn't really happening very much. Okay. Right, now with asexual and sexual, right, what I'm going to do now is I'm going to go through the 3 examples, right, that they talk about on the syllabus. Okay, now some organisms do both. Okay, so the first one is Plasmodium vivax, which is your malarial parasite. Right, now again, I'm not going to go through the whole process of the malaria cycle, right, but malaria does asexual and sexual reproduction depending on where it is. Okay, so think about the cycle. It gets injected in from the mosquito. Okay, it goes to the liver, it goes to the red blood cells, it's then in the blood, and then it goes out again. Asexual reproduction is in the human, right, when it is actually in the liver and the red blood cells, right? So when the plasmodium is in the human, in the liver, in the red blood cells, that is when you've got asexual reproduction. Sexual reproduction occurs in the actual mosquito itself. Okay, and that is then all to do with a temperature change. Okay, so mosquito bites, sucks. When that then sucks out, what happens then is it changes the plasmodium to then do sexual reproduction. Okay, another example, fungi. Right, so fungi, right, they do asexual reproduction when the conditions are good. They do sexual reproduction when the conditions are harsh. Strawberries, asexual reproduction when they have runners. So if you put strawberries in your back garden, you should never ever plant them straight into the ground. Because what a strawberry does is it just puts a runner down, the runner goes underneath the ground, pops up somewhere else, and that is asexual reproduction. Sexual reproduction is then from the flowers. Okay, so that is then certain animals, again, these are particularly named on the actual syllabus itself as living things that do sexual and asexual reproduction. Right, brain. Okay, so there's the brain. Okay, then there's a breakdown of it. Okay, now I know that we've gone through this in certain sessions a long time ago. Okay, right, so we start at the top. Cerebrum, okay, is your voluntary actions. Okay, thought, vision, speech, memory, problem solving. Pretty much what you've got to do is you just got to remember it. Okay. Cerebellum, muscle coordination. Okay, so that is the bit right, the soft bit right at the back of your head. You bang that, you're in trouble. Medulla oblongata. Okay, so remember I talked about that previously and I said it's separates only. Okay, that is then your involuntary. Okay, which is basically the top of the spinal cord. Heart rate, breathing rate. Hypothalamus is one of the big control centers, okay, and homeostasis. So when the blood goes around the body, the blood goes through the hypothalamus and the hypothalamus detects what is there. It'll detect oxygen levels, glucose levels, water levels. And when the hypothalamus actually detects it, what it then does, it'll then send nerve impulses, and particularly it'll have a chat with the pituitary gland, and the pituitary gland will then send out a load of hormones. Okay. Right. Anybody got any questions about the brain? Okay. Now, I have to admit, I personally forget. I find it difficult trying to remember all these things, but you guys are way smarter than me. Okay. Right. How do we study it? Okay. Now, there's a number of different ways in which we can study the brain. Okay. Now, or difficulties rather. So difficulties is the fact that the brain is incredibly complex. Okay. Studying the brain is bad ethical wise. Okay. Because is that the right thing to do? Studying the brain is difficult with people that have got damage and disease. And the brains of everybody is also very different. Okay. So often the way they'll do the kind of the studying of the brain is they'll ask for volunteers. Okay. And what they'll also do is they will particularly ask for volunteers that have actually got brain damage. Right? Because if you think about somebody that's got a stroke, right, and certain aspects of their kind of ability to do things has diminished, take a picture of their brain, you can then work out which parts were responsible for that. Right? I can remember seeing videos, not really weird videos, of monkeys. All right, I suppose it is pretty weird actually. Where what they did is they cut the skull off, top of the skull, and they had the head kind of sticking out. Right, and then what they got is they got the electrified needles and they were jabbing the monkey in the brain. Okay, and then when that monkey had certain parts of the brain was actually stimulated, the monkey was kind of moving its arms. Okay, And I can also, when I was at university, remember the professor talking about the same sort of thing with humans. Okay? Where what they actually had is they had parts of the skull taken off, right? And then they'd actually put electrical impulses into the brain and it would then stimulate memories, right? Where people would be kind of lying there and they'd have electrical impulse and they'd start singing a song from when they were 3. Okay? And that is then another way, but obviously that's pretty unethical to actually be able to do those things. Okay? Right. Decay, required practical. Okay? So what I'm going to do is I'm literally going to give a minute, right? Maybe 2 minutes. All right. And what I want to know is I want the full method for the decay required practical. And I've written on the screen there, okay, is investigate the effect of temperature on the rate of decay of fresh milk by measuring the pH change. Okay, so for this one, I want numbers. I want to know the chemicals that are involved, and I want to know why. Okay, so I suggest that what you actually do, just to impress me after that last required practical, I suggest on a piece of paper, you write down the method for the decay required practical. All right, Daniel? Can you write it? Just write it, because tomorrow you might get asked to write it. So if you write it down now, right, and then if I say anything different to what you've written, okay, then add it in. Zara, are you all right with this one?
Yeah.
Yeah. Boardi, are you okay with this one?
Yeah. So I want volumes. I want names of chemicals. Okay. Temperatures. Why does it happen? Right. Go for it. Right. So I'm going to write down what somebody says to me. Okay. Farhan, go for it then, please.
So you'd need like different beakers of milk, right? Then you'd get water bath.
All right. So different beakers of milk.
Yeah.
Okay. Yeah. Carry on.
Oh wait, no. Wait. Effect of temperature on the rate of the. So then you get water bath and you put them to different temperatures, like different intervals.
So I've got a water bath. Okay. And I'm going to put the beakers into my water bath
Mm-hmm.
all at the same time.
You can do them one at a time. Doesn't matter.
Do one at a time. Well, let's just put one in then. Okay. So we'll put one beaker in. Okay. Right. So I've got a beaker in a water bath.
Yeah, and then you put your pH indicator in.
I'll
No.
put a pH indicator in.
Wait. I'm pretty sure.
I'm just going to put a pH indicator in, and that's it. What do you mean? I don't understand what you mean.
That is milk.
Right? Do you want me to move on?
Yeah, I don't really know this one. Not gonna
Right. Right. Well, everybody needs to know both of these because there's only two. Okay. Zara. Okay. Apologies about my stupid camera zoomed in again. Okay. Zara, tell me the method, please.
Okay, so you get 5 centimeter of milk
5 centimeter cubed of milk.
in one test tube.
Okay, in a test tube.
And then in another test tube, you get lipase and label that test tube with like L so you know which one's which.
Okay, I'm going to label it L. Yep.
And then in the milk, you're going to add I don't know how to say it.
Right. Phenol. I always call it phenol per valine. Phenol per valine. Oh, I spelled it wrong as well. Thal. E-I-N. Yeah, go on.
And then in that same test tube, add in like sodium hydroxide, 7 centimeters of that.
Sodium hydroxide. How much? 7 centimeter cubed.
Yeah.
Okay.
And then it'll go pink from like white to pink.
So it'll go from white to pink. Oh, the milk could go from white to pink. Okay.
Yeah.
Yeah.
And then after that, put both test tubes in a water bath at 30 degrees and put a thermometer in the milk, and when it reaches 30 degrees then turn it off.
Okay. Right. But if the water bath's at 30 degrees, we could just leave it in there for a while, couldn't we?
Yeah.
We'll put a thermometer in. Okay, good. Okay. So stupid camera. Go on, carry on.
And then after they're both at 30 degrees, get a stopwatch ready and add 1 drop of the lipase into the milk and time how long it takes to go from pink to back to the white color.
Okay, so stop clock, and then you add how many drops of lipase?
Just 1.
1 drop lipase, and then you time how long it goes back to milky color.
Yeah.
Okay. Right.
And then,
Go on.
and then repeat this for like different temperatures.
Repeat for different temperatures. Right. Okay. Now that's good. I'm just going to ask you one double quick question, Zara. You know, and you're so close on this one. You know NaOH,
Yeah.
sodium hydroxide. Okay. What sort of pH is sodium hydroxide?
13.
That's 13. That's a solid alkali. That is oven cleaner. Okay. Sodium carbonate solution. Okay. So it's not sodium hydroxide, it's sodium carbonate.
All right.
All right. Right. Now, you're on the right track. Okay. So I'm just going to put my camera down. Okay. Adam, right. What pH does sodium carbonate actually make the milk?
Would it make it more acidic?
Nope, doesn't. Sodium carbonate is actually going to make it alkaline.
Okay, all right.
Okay, but I'm going to tell you a number in a minute. Okay, right, Daniel, right, the sodium carbonate makes the milk become pH 8.3, or it makes it greater than pH 8.3, right? Why does the lipase make it more acidic?
Is it because it breaks down the lipids into fatty acids and glycerol?
Good. Okay, it's the fatty acids that then change the pH. Well done. Right, now I'm going to put up the method. Okay. Right. So, Zora, you are pretty much most of the way there. Okay. We just need to remember kind of a few maybe additional numbers. Label a boiling tube lipase. 5 centimeter cubed of lipase solution. So you got boiling tube with 5 centimeter cubed. Okay. Another one's got milk. Now you should use full fat milk. Okay. So you shouldn't just use that red stuff. You know, that stuff's like dishwater. Okay. You shouldn't use that stuff. You should use the blue milk, full fat milk. Okay. Add 5 drops of phenolphthalein. Okay. Now there is another one that you can use, which is called creosol red. Okay, but the main one that is used is phenolphthalein. Okay. 5 centimeter cubed of milk in the milk one, and then you add to that 7 centimeter cubed of sodium carbonate solution. Okay, so in that boiling tube now, you've now got 12 centimeter cubes of liquid. Make it greater than pH 8.3. So the sodium carbonate makes it alkaline greater than 8.3. The solution should then go kind of a pinky purple color. Okay. Both boiling tubes into the water bath. So Zara started off saying 30 degrees C. Okay, that works. Now, if you think again, Zara, if you put 1 drop of lipase in there, okay, you're not talking about very much stuff. Okay, so personally, I would go for 1 centimeter cubed of lipase. Okay, as soon as you put that 1 centimeter cubed of lipase in, you start the stop clock. Stir the contents until it decolorizes. Okay, so literally the pinky purple color goes. Record in seconds, then repeat different temperatures. Record your results in a table and then keep repeating, or to make it more accurate, repeat each one so then you can plot the graph. Okay. Right, now we need to think about some control variables, right? So don't mess. What are we measuring? Right, what are we measuring? Adam, what are we measuring in this?
The time taken for the solution to neutralize.
No, it's not neutralizing. Neutralizing is pH 7.
To acidify
Okay.
them.
Do what, sorry?
To acidify.
It's not acidify either. Okay.
Less alkaline.
So what it is what is that 8.3? What does that 8.3 mean? Anybody, nod at me if you know what that 8.3 actually means. Go on, Zishan.
It means it's an alkali.
Yeah, it does mean it's an alkali, but why on earth am I going to put 8.3 on there? Okay, I could have put...
That's the pH of it.
Yeah, it is. That is the pH of it, but why is 8.3 significant?
I don't know.
Is it because it turns colorless when it's below pH 8.3?
That's exactly it. So 8.3, well done, Daniel. 8.3 is the pH that phenolphthalein changes color. Okay, so that is then why that is significant. Okay, so what you do is you put in the sodium carbonate. Okay, then what you do then is it goes greater than 8.3. And then when you then start adding the milk and the lipase is producing, like Daniel said, the fatty acids. The fatty acids then gradually reduce its pH to become acidic. And then when it gets to basically 8.2, that is then when it changes color. So it's nothing to do with neutralize. It's nothing to do with pH 7. 8.3 is the number that makes a difference. Okay. Right. So don't mess in class. Dependent, what we measure is the time that it takes for the phenolphthalein to go from clear to colorless. Don't mess in class. Independent, what are we going to change? The temperature stays the same. Control. Okay. Zishan, give me a couple of control variables, please. Can't hear you.
You keep the same volume of milk, volume of the lipase, and the volume of red. I forgot it. Just the volume of phenolphthalein.
Yeah, the volume of the same volume of sodium carbonate. That's good. Okay. Right, Adam, give me another one.
The volume of lipase added to the solution.
Yeah, so we've got all the volumes. So we've got volume of milk, sodium carbonate, lipase, all the indicators. Right, we need another control variable. Think about your disappearing cross.
Concentration of the sodium carbonate solution.
Yeah, that works as well. Okay, what's one of the control variables for a disappearing cross that's a bit different? The person. Okay, because it's not necessarily really obvious with the actual color change, even though it most of the time is. It needs to be the same person as they're making the decision. Okay.
All right.
Right. So That's it. Lipids, lipase makes fatty acids and glycerol. Okay, that's good that the acidity comes from the fatty acids, but don't forget your 8.3. Okay, so there is no neutral, there is no neutralization. Well, there is neutralization, I guess. Okay, but it's not like neutralization as we imagine it. That's good. Right, there's two typical graphs that you might get. Okay, the left-hand graph is rate against temperature. Okay, the right-hand graph is time against temperature. And obviously what you can do is you can see the different shapes of the graphs. Okay, so again, just be aware of what they're actually after. You got to think that the faster it goes, right, then the more optimal the temperatures are. Right, now you're all dead good at that. You can all read graphs. There's not an issue there. Okay, right, now they're the two required practicals. Okay, now what we need to do is we need to go through a load more kind of theory. All right, so sustainable fishing. Okay, it kind of makes sense, really. Right, so what you got to do is you got to maintain the amount of fish in the sea. Right, so when you take fish out, okay, then what you got to do is you got to make sure that you're not taking too much out, so that the number of fish in the population decreases. Okay, now how do they actually do the sustainable fishing? What they'll do is they will control the net size and they will give people fishing quotas. All right, so the idea of the net size is the size of the holes. Right, so then people that are then actually doing the fishing, they're only allowed to catch fish of a certain size. Okay, and then the smaller fish will then escape through the holes, and also give them fishing quotas. Right, so when you actually are a fisherman and you go to the port, right, where the fish is, you are then only allowed to put in a certain amount of weight of fish. Okay, right, so the way that government control it, okay, is they give quotas, they will inspect, right. Bycatch is all the extra kind of like living things that are in there, and you're only permitted in certain places in the actual oceans themselves. Okay, there's certain sections that are no-goes. Okay, so sustainable fishing is keeping or only catching enough fish so that the population isn't long-term affected. Food security. Okay, so what I've got here is this kind of diagram, and again, it's something that you can kind of look at later, is food security is important, all right? And there's kind of four things here that are interlinked with food security. We've got access to food, the availability of food, using the food, and then the stability of it. Okay, so food security is basically, can somebody get hold of the food? As well as the availability is kind of getting hold of the food as well. Using the food, and then stability is then stuff over time or keeping it kind of safe. Okay, right, mycoprotein, right, is then obviously a protein that is mentioned again on the syllabus. Okay, and you'll be aware of it if any of you do kind of food tech or anything like that. Mycoprotein, right? So mycoprotein, if you go to the supermarket, it's kind of those white globby things that are supposed to look and taste like meat, but they don't. They're kind of a bit like, I know, some strange alien texture stuff. And what mycoprotein is, it's usually Fusarium fungus. Okay, and literally what they do is they brew it a bit like beer. Okay, so they ferment it, right, with glucose, and then what they'll do is they'll do some certain processes. They'll steam it, chill it, and freeze it, right? And those different processes then give it its meat-like texture. Okay, so they have a massive, great big vat, a fermentation vat, right? The fungus is in there growing, eating the glucose. Then what they'll do is they will then spin it to centrifuge it. They will then chill it to then take it out as a solid mass, and then they can then take it away. So it's pretty quick. So that's mycoprotein. Right, trophic levels. All right. Now, again, trophic levels is biology only. I've kind of written out at the top there. So this is then our food chain. So we usually start off at the bottom, don't you? Producers. Okay, so producers is your plants. 2, primary consumers. Secondary, tertiary, quaternary. All right. Now, those words to me are pretty obvious. What's the first school you go to? Primary. Then you're in secondary. College is tertiary. University is quaternary. Okay. The different levels of kind of education that you're also going to go through. Okay. They are your trophic levels. You've got biomass. Okay, so bio numbers is kind of on everybody's, right? But I think that was reasonably straightforward. Biomass is then where you are literally just talking about the mass that is involved in a food chain. Okay, now a biomass pyramid is always that shape. Okay, and they normally say that something like only about 10% of the energy goes from one stage to the next. So here is then a typical kind of transfer of biomass kind of diagram. So you start off with the producer and there's 1,000 kilograms of it. When it goes to the primary consumer, literally only 10% of the biomass carries on to the next trophic level. 10% again, 10% again. So if you think about the efficiency of this, it's pretty horrendous, really. You start with 1,000 kilograms of plant, and the strange-looking wolf at the end there, there's only 1 kilogram left. Okay, where does all that energy go? Respiration, feces, waste, carbon dioxide. All right. So if they do talk about anything to do with trophic levels, remember, only 10% goes up each trophic level, and the rest of it is then lost, usually by kind of feces, right? Because if you eat something, you don't absorb everything out of it. You use it for respiration, so therefore carbon dioxide, water, and all those kind of things are then given out as well. Okay. Right, again, speciation, we did kind of talk about this before. Okay, so speciation is then when a founder species becomes other species. All right, and then just remember what a species is. A species is unable to have fertile young. Okay, right, so speciation is the process of going from a species to other species that are incapable of producing fertile young. Okay. Again, another name that is mentioned on the syllabus is Alfred Russel Wallace. Okay. He is the guy that came up with the idea of speciation. Alfred Russel Wallace. His name is written in black and white on the syllabus and has never been on an exam. He's the speciation guy. Okay. Right. The eye. Okay. I'm not going to ask you to do abacadabra, all those things. Oh, I suppose you could do. Right, but I'm not going to. Okay. Right. I'll just go through them. Okay. Right. So the eye. We need to know the structure. Okay. So if we start off with the retina. Right. The retina, light sensitive cell layer. Okay. What are the names of the cells in there? Okay. We've got rods and cones. Okay. The optic nerve is then the nerve that then goes out at the back of the eye. Okay. Then we know what that bit there is called. Right at the very back of the eye, it's kind of where the optic nerve goes out, but it's actually got a name. Anybody nod if they know what it is?
So is it the blind spot?
It is the blind spot. Well done. Okay. So what you've got in the back of your eye, because it's all kind of concertinaed up there, all right, that is then your blind spot. So, you know, sometimes you can kind of be looking at something and then a certain part of your vision disappears. Okay, that's then because the light is then shining directly onto your blind spot. But because you've got two eyes, it is never obvious and your brain can work it out. That little dimple there is your fovea. Okay, I never know how to pronounce that. Fovea. Fovea. Okay, that is then the part that is your kind of main focal point. So when the light comes in, that is then where it focuses. And that contains the most cone cells. Okay, cone for color, cone for clarity. Okay, so when you're actually looking at something, that light is going onto your fovea, and all the way from the outside, it gets blurrier and it gets less color. So going this way, you're going to have more rods, and going this way, you're going to have more rods. Okay, right. The other bits, you've got the cornea, which is basically a protective layer, right? If you get conjunctivitis, conjunctivitis is kind of like an infection of the outside of your eye. Pupil is the black hole. Lens is obviously the part that actually focuses the light. Ciliary muscle and suspensory ligaments, they are then the parts that then stretch and sort of relax the lens. Okay, right. Dim light, how do we actually do it? Okay, so there's some kind of key wording in here, right? So if it's like dim and you need to open your pupils a little bit more, you've got two sets of muscles. The first muscle, the first muscle, the radial muscles. So kind of makes sense, right? That when it's dim, these radial muscles are lines that then go to the kind of the center of the pupil. And if a radial muscle contracts, the pupil gets bigger. In bright light, what happens is the ciliary muscles contract. So they are kind of circular around the outside of the actual pupil. And when they contract, the hole gets smaller. Okay? And if you actually look at somebody's eye, what you can do is you can kind of see these kind of muscles that are in there. Right? Not particularly very clearly, but you can see them. Right, myopia and hyperopia. Okay, now the main thing you've got to remember these is you've got to remember what the actual myopia means and what sort of lens you're going to have. Okay, so myopia, short-sighted. Okay, which means the lens is too thick and the image is there. So myopia, short-sighted, and that's then when you have those whopping great big thick lenses. Okay, so what are they? Concave. Okay, concave means it goes in at each end. Okay, so myopia, short-sighted, you need a concave lens. Hyperopia is long-sighted. Okay, and with this one, you then need a convex lens, okay, which is the one that most people then tend to have. Right, from about the last 10 slides, anybody got any questions about anything? Okay, just wording here now, wording. Right, water. Right, so when we talked about homeostasis before, homeostasis is the maintenance of the body for optimal conditions for enzymes. Right, we talked about glucose, which is your insulin and your glucagon. Right, and again, insulin is on everybody's, glucagon tends to be more on higher, and it is more asked about on the separates paper. Okay, remember glucagon. So glucagon is you use up sugar, so therefore you produce glucagon from the pancreas, which then turns glycogen into glucose to go back to homeostasis. Okay, right, water. If you've got too little water in the blood, we've talked about the hypothalamus so far, it detects the amount of water. Okay, so I can feel it at the moment, right? I feel like I need a drink. My hypothalamus is telling me I haven't got enough water in my body. I then would produce, or I am producing, ADH. So therefore, more water is absorbed by my kidneys. So therefore, less water is lost. Then what happens is we have our negative feedback, which then takes it back to homeostasis. We've got two Z-channels now. Okay, so controlling water. Too little water, hypothalamus detects. Pituitary gland releases ADH, antidiuretic hormone. More water is reabsorbed, less water is lost. Blood levels then go back up again. Okay, Kidney is also part of that. So what the kidney can do is the kidney will sort of filter out the water. Okay. Now, they also could ask about dialysis. So dialysis is where you literally have your blood getting pumped through a dialysis machine that then removes the urea. Okay, so the two treatments for kidney failure, organ transplant or dialysis. Okay, and I think with dialysis, you have to sit on like a dialysis machine for, I don't know, 5 hours a day, 3 times a week. Okay, so that can't be particularly very nice. Heat. Okay, so body temperature. Right, so again, we've kind of got two sort of systems that are involved in this. All right, so we've got, if our body's temperature is too high, we do vasodilation. Okay, think about it. Vaso is kind of your vessels. Dilation means get bigger, which is this one. Okay, so vasodilation means the little capillaries underneath your skin get bigger. When they get bigger, more blood goes in. You go pink in the face. You can then lose heat by radiation and convection. When your heat is too low, vasoconstriction, vessels constrict, which is this one, where you'll then go paler because all the blood goes away from your skin. Okay, right, now we've talked about the hypothalamus already, okay, which kind of detects everything. Okay. We've sort of talked about that. Right, cloning. Okay, so this is now our kind of last little sections. Okay, so what we've got now is we've got cloning and we've got protosynthesis and mutations. Right, now again, we've talked about cloning in kind of these sessions before. Right, so what we've got is we've got cloning. A nucleus is removed from an unfertilized egg. Okay, the nucleus from the one that you want is then inserted. Electric shock is there to stimulate. The embryo is then genetically identical. Then you get a ball of cells and they're reintroduced into the host womb. Okay, so the famous one on that one is Dolly the sheep from Edinburgh University, which was then the very, very first cloned kind of large animal. Right, so you got an egg, take the nucleus out of it. Put the nucleus in from one that you want, electric shock, it'll then develop, put it back into the host. Other cloning techniques, so we've got kind of like tissue culture, cuttings, and embryo transplants. Okay, so the first two of them mostly plants. So tissue culture is when you literally get a few kind of cells. Cuttings, chop one off, chop a branch off or whatever it is, and then you can then grow it into a full plant. And embryo transplants is then what we just talked about previously. Right. Mendel. Okay, right. Last tiny little thing then. I'm worn out. Are you worn out, Adam? I'm worn out as well. Brian's well tired, look, you can see. Okay.
I don't even sleep anymore.
You don't sleep anymore?
No, I just sleep like short out, long days and short nights.
Yeah. But just think what's going to happen in about a week and a day.
Yeah.
You have to make up for it all then, won't you?
Mm.
You have to sleep 24/7 for like 10 weeks.
I'm looking forward to it.
Oh, I'm not surprised actually. Right, so DNA structure. Okay, so what we've got is we've got our base pairs. Okay, AT, CG, adenine, thymine, cytosine, and guanine. Okay, so what they do is you should know the kind of the base pairings. Okay, they are then the ones that kind of link together. 4 possible mutations: duplication, deletion, inversion, and translocation. Okay, so duplication, got 2 B's. Okay, so it's a possible mutation. Some mutations can be beneficial, some are not. Deletion, B's now gone. Inversion, everything kind of swaps. Okay, and you've got to think that every single time one of these happens, it creates a different protein, right, to then make either a good thing happen or a bad thing happen. And translocation, okay, is then where we've got kind of that capital E has gone over to that side there. Right? So the 4 main groups of mutations: duplication, deletion, inversion, translocation. Right, final thing is protein synthesis. Okay, so protein synthesis, what you'll do, I'll do another diagram in a minute. We've just got to remember the differences between DNA. So DNA has A, T, C, and G. mRNA has A, U, C, and G. Okay, you just got to remember in mRNA, okay, the T becomes a U. Like it's uracil. And then each one of those kind of codons makes an amino acid that then makes a chain. Right, now I'm going to show you the protein synthesis diagram. Okay, now it's quite complicated, okay, but you're all with it. Right, so protein synthesis. What it does, it starts off with transcription. Okay, so what happens is the DNA unravels. Then what it does, it makes a strand of mRNA in the unraveled DNA. Okay, that strand of RNA, that is then called transcription. Okay, so you're transcribing something. That strand of mRNA then goes out of a pore in the nucleus and heads towards a ribosome, which is these here. So the ribosome here, this green thing, the mRNA is on it. And then you've got tRNA, which is transfer RNA, brings things called anticodons. So an anticodon is literally 3 bases. So the tRNA brings the anticodon, and 3 of the bases in a protein or an mRNA is a codon. So those 3 anticodons link up to the codon. They then link, and then what happens is the mRNA kind of moves its way along. Okay? And as the mRNA moves along, what it does is it produces an amino acid chain, and the tRNA entering the mRNA is called translation.
So,
Yeah.
so the anticodon, does it take the exact same bases as the ones in the mRNA, or does it take the one that corresponds?
It's the corresponding one.
Okay.
Yeah, so on the anticodon, if the anticodon had a G, okay, then on the codon, it would then be a C.
Yeah. All right.
Okay? So it's the opposite one. Okay, and eventually what you'll have is you'll have your newly formed protein. Okay. Done. Done in. You realise something? I normally don't ever talk. I've just talked for 3.5 hours. Long time. Right, Daniel, have you fallen asleep?
Nah.
You're just kind of in your PJs now, are you? Right. Boaji, are you okay?
Yeah.
Yeah. Right. Now, I've obviously talked for a very, very, very long time tonight. Now, is there anything in particular that anybody is worried about? The only one I'm worried about is that first required practical one with the plants. All right. So every single one of you, right? I know it's quite late now anyway. right? You all need to go away. You need to make sure you know that one inside out. Okay, every single little control variable and every single thing, right? So remember, it also grows the longest in darkness, because basically the plant is growing to try and find a source of light. Okay. Zara, are you okay?
Yeah.
Right, now what I'm going to do is these PowerPoints are already up on Google Classroom. So have a look through it if you want. And then this entire session was then recorded. Okay, but it takes about an hour for my computer to actually turn it into anything. All right, so what I will do is I will then put that up a link to YouTube, right, so that you can have a look through things if you need to. Okay, does anybody have any questions? Zishan, when am I seeing you again then?
The camera on this device doesn't work, sir.
What, sorry?
The camera on the device I'm using right now doesn't work.
All right, okay, that's fine. All right, no, I didn't mean when am I going to see you again. When's your next exam? Is it Friday? Yep. So Thursday night, chemistry paper 2. All right. Right. Unless anybody's got any questions, right? Go and get some tea. Okay. Thank you, everybody. You're all completely amazing as always. Okay. Just learn that required practical.
Thank you, sir.
All right.
Thanks, sir.
Yes. See ya.
Bye, sir.